Hi everyone. I hope I'm perfectly audible. Yes, I hope I'm perfectly audible. So hi everyone, welcome to Unacademy NEET English. So finally we are meeting for the session, and that too after a long time. You guys have requested us to start the series again. So this series is specially for the class 11 syllabus. Those who think that those who are still in class 11 and they think that they have wasted their time, this series is for them, because here we are going to complete the class 11th syllabus first. Okay, right? So I have completed majority of chapters. I think only 2 or 3 chapters are pending, which are not done in game of NEET 1.0 or 2.0 or 3.0. Rest everything is done. I think 1 or 2 chapters are there, but otherwise everything is there on this channel. So when you check this channel, you will get all the content that is sufficient for your NEET 2026. Don't worry about that. Okay, don't worry about that. So today the chapter that we are going to start is Human Physiology, and this is the first chapter. This is a unit that we are going to start, Human Physiology, and the chapter that we are going to start is, you know it very well, that is the breathing and exchange of gases, right? So which chapter are we going to start next? Today it is the breathing and exchange of gases, and after this we'll start with the locomotion and movement. So it is as per your request. I asked you people, what do you want to proceed with, plant physiology or the human physiology? You people selected, majority said that it human physiology, so I am here. So let's start. Good evening guys, let's start the session. And moreover, here you people can see these are the links in the description box here, right? These are the links in the description box here, Unacademy revision for Unacademy revision test series, right? So those who are studying by themselves, right, those who are basically just doing the self study, for you this is very important. Test series is very important. You really need to analyze your performance, and here you are going to get very good paper as per the PYQs, completely NCERT based, and you are going to love it. And for the breathing and exchange of gases, the practice questions will be uploaded here. You can just check it out, right students, and you can join our official Telegram group. So let's start the session. Okay, let's start the breathing and exchange of gases, one of the easiest chapter. Okay, ma'am, if I start now, can I get seat? Beta, like from where are you starting? You know something or you don't know something? Firstly tell me this. Because if you think that, ma'am, I don't know anything, I don't know anything from 11th, I don't know anything from 12th, and now onwards I'm going to start, then obviously you will not be able to complete your syllabus. Okay, if you have done something, if you have studied something, if your revision is not up to mark, or if only few chapters are pending, then obviously for sure, if you will give your best, then you can complete your maximum syllabus. So I am not here to give you the false hopes that no worries, if you will start now you can get the seat, because that is literally not possible. Students, they give their 2 years, 3 years, sometimes 4 years or 5 years for this particular exam. So if you think that last 2 or 3 months will be more than sufficient just to complete everything, not possible. Yeah, but yes, if you have the idea of your syllabus, you have studied earlier, but from quite some time you were not studying sincerely, then definitely if you will follow a proper strategy, then definitely you will crack it. Okay, so I'll be very honest here. So let's start the chapter. So here you guys can see the name as well. So what's the name? Breathing and exchange of gases. That's the chapter name, breathing and exchange of gases. The first chapter from the unit Human Physiology. Okay, so let's get started. So when we talk about the breathing, what exactly is the breathing? I'll relate it with the food. We take the food, right? We take the food. Why? Because we need the energy. Isn't it? We need the energy. We know that whenever we eat something, right, the complex food, it will break down into the simplest one. This is what digestion is. Then you know that the absorption of food will occur, assimilation will occur, all that things will occur. So we all are aware of it, isn't it? We all are quite aware of it, that all the things that happen, we take the food, our body is going to break that complex food into the simplest one. This is what digestion is. Then absorption will occur, assimilation will occur, waste will be ingested out. This is what we know. But why do we need that food? We need that food for the energy. Our body, it needs energy, right? It requires energy for its metabolism, right? For its activity. So we all are aware of it. Now, what's the thing? Okay, you have the food in your body. Now what? We know that if I talk about the basics here, right? If I talk about the basics here, so we know that if I talk about the carbohydrates, we know that the basic thing here is what? The glucose, isn't it? Yes or no? The glucose, like all the complex form, they'll convert into the simplest one. We will have the glucose. So what will happen? Its oxidation will occur, isn't it? Its oxidation will occur. By using oxygen, we are going to break the glucose so that what will happen? So that energy can release. So that energy can release. Yes or no? Yes or no? And what's this process? What's this process? This is actually respiration. Yes or no? What is it actually? It is actually respiration. Yes or no? This is what respiration is, the breakdown of this glucose, right? The breakdown of this carbon-carbon bond, right? So basically the oxidation of glucose occur here in multiple steps by using enzymes so that energy can be released. It can be trapped in the form of ATP. So we need this thing to oxidize the glucose. We need oxygen. So when we talk about the respiration, in the respiration we talk about the breakdown of food so that we can get the energy. And as I said, I'm repeating this again, in multiple steps by using enzymes, and then that energy will be trapped. So from where are we getting this gas? From where are we getting this oxygen? So this is going to be your first step, that is breathing, also known as pulmonary ventilation, which is basically what? Which is basically the exchange of gases. So sometimes we use these terms interchangeably, right? Sometimes we use these terms interchangeably, but actually it is the breathing that is the very first step of the respiration. So that is how I started. So we need energy, we need food, we need these compounds, so we have to break it so that our body can get the energy in the form of ATP. So when it comes to the respiration, which is giving us energy, here there are some steps. You guys can see these 5 steps here. So the very first one is here what? The very first one is breathing. What is it? It is breathing. Are you getting my point? So breathing or pulmonary ventilation. What's that? It is breathing or pulmonary ventilation. Are you getting my point? What is it? It is breathing or pulmonary ventilation. So it is the process by which atmospheric air, it will be drawn, right? It will be drawn in, and the CO2 rich alveolar air will be released out. So this is what we are talking here. So this is what breathing is. Okay, the very first step of respiration. Anybody having any doubt? Yes? Anybody having any doubt? So this is what you need to understand. Next. Now, once you have inhaled the air, then what will happen? It will go to your respiratory organ. What is our respiratory organ? We have the pair of lungs. Now, in the lungs, the primary site is the alveoli. Okay, one by one we will reach there. We will discuss it in detail. So now in the alveoli, there will be the exchange of gases. How come? So from that alveoli, because that lungs, they are having abundant blood supply, right? They are highly vascularized structures. So when it comes to the alveoli, the exchange of gases will occur. So oxygen will diffuse into the blood and CO2 will diffuse into the alveoli. So here you guys can see diffusion of gases across the alveolar membrane. This is the second step. Then obviously blood will transport that gas, the diffusion of oxygen and CO2. So blood will carry that oxygen to where? It will carry that to the tissue, right? So the tissue can use it. So the cells there can use it. They can perform the respiration. So diffusion of oxygen and CO2 between blood and tissue will be there. So because blood will have high partial pressure of oxygen, it will go to tissue. Tissue is having high CO2, so it will pass to the blood. So that is how the diffusion will occur here. And finally, oxygen will be utilized by the cells for the catabolic reaction, for these breakdown reactions. So what is the meaning of catabolic reactions? In which breakdown is there. In which what is there? In which breakdown is there, and there is the release of carbon dioxide. So this is the cellular respiration that we study in the plant physiology, right? Under the chapter respiration in plants, where you talk about, you know, everything there, like yes. So what do you discuss there? You people talk about? Yes, please. What do you discuss there? So in the respiration, we'll start with the first step, glycolysis, and then for aerobic, we go for the Krebs cycle and the electron transport chain. So this is what you need to keep in your mind. These are the 5 steps of the respiration. I hope it is clear to everybody. Am I clear? I hope it is clear to everybody. These are the 5 steps here. Is that clear? Okay. So now, obviously, we are going to cover the NCERT. If somebody says that no need to, right? If you go offline as well, and people say no need to, or even in online that no need to study the NCERT. No. This is the book that we have to understand. But that doesn't mean that you have to stick to that book only. Actually, to understand many concepts given here in this book, you have to go beyond NCERT, and we'll do the same. But ultimately, you have to understand this book, you have to understand each and every detail here, and then only guys, we can further go ahead. Understood? Okay, so the next topic is the respiratory organ. So this is what we have to study. So before starting the respiratory organ, let's talk a bit about the animal kingdom. Okay, not talking about the subkingdoms here directly. Let's discuss some phylums. You know that we belong to the animal kingdom, Animalia or animal kingdom. So we have the 11 phylums here: Porifera, Cnidaria, Ctenophora, Platyhelminths, Esculminths, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, and then finally we have Chordata, isn't it? These are the phylums that we have. So when we talk about the animal kingdom, overall we know that life started in the water, right? This is what we know, right? So once we'll after this 11th part, whenever we'll start the game of NEET, okay, game of NEET, so I will start with the evolution. Okay, I'll start with the evolution. I'll simplify evolution for you. So we know that the life, it started in water. So basically now you are talking about the animal kingdom. You know many things, that obviously organisms, they are eukaryotic, organisms, they are multicellular, and when we classify them, how do we go? We move from simplicity to complexity, right? Whenever you talk about any of the classification, how do you go? You go from simplicity to complexity. That is how we move. So when we talk about the simplicity and the complexity thing, like simplicity, you are talking about the poriferans, just having cellular level of organization, having no proper respiratory organ, right? Living in the water. Okay, the gases exchanges through the general body surface, and obviously it will occur through the diffusion only. Okay, so when you move from porifera to chordata, what exactly is happening, right? The organisms, they are becoming complex, their body is becoming complex. Organisms, they are basically, the advancements are there in the organisms. So when we move from porifera to chordata, so we know that the advancement will be there, the complexity will increase, so we are aware of it, isn't it? Like here you will see cellular level of organization, tissue level, organ level, then organ system level is there. We are well aware of it. So now why are we discussing here in the breathing and exchange of gases part? So it's simple. So when you see the different type of organism, as per their habitat, as per their level of organization, you will see that they have different types of respiratory organs as well. For an example, if I talk about the poriferans, right, where you know, the cellular level of organization is there, right? They are sessile organisms. So there you will see that respiratory organ, a dedicated respiratory organ is absent, right? They don't have the respiratory organs, but instead the respiration will occur through the general body surface. So these are the things that we really need to know. Are you getting my point? So when you talk about what? When you talk about the porifera, so from porifera to esculminths, you are simply going to remember this thing from porifera to esculminth. So this exchange of gases is through general body surface. Are you getting my point? So exchange of gases is through general body surface. Are you getting my point? It is through general body surface. That is how the exchange of gases is occurring. So next to it is annelida. Obviously in the annelida also, if you ask me that is there any dedicated respiratory organ? No. Here you will see that moist cuticle is there. Annelida, you have the example of earthworm here. Isn't it, guys? You have the example of earthworm here. So in the case of Annelida, what will you see? That they will have the outer covering again. So here also you will talk about the moist cuticle. Then we have arthropoda. What do we have next? We have arthropoda. So arthropoda is exactly what? It is the largest phylum of animal kingdom. What's that? It is the largest phylum of animal kingdom. What? Your arthropoda. Am I clear? Largest phylum of animal kingdom it is. And here in this phylum, for the first time, you are going to see that there is the dedicated respiratory organ. Right? Properly respiratory organs we will see in this particular phylum. Like if I talk about the insects, you take the example of your cockroach. Right? So insects, they have the tracheal system. Yes or no? Important it is. Very important to remember. Yes or no? So what do they have? They have the tracheal system. You have the example of cockroach there. Right? Then let's talk about the arachnids. Like class Insecta, class Arachnida. So here you have the example of spider. So you will see here what? Book lungs or book gills? Anybody? Book lungs or book gills? Again, a very, very important question here. A very, very, very important question here. So it is book lungs that you are going to see. Right? It is the book lungs that you are going to see. So these are the examples that we have. Yes or no? Yes or no? Yes or no? Done. Then in the aquatic arthropoda, aquatic arthropods, what do you see? You will see book gills. And here if I have to give you the example, so that is going to be limulus, king crab, living fossil. Are you getting my point? So that is what you have to remember. Okay, that is what you have to remember. Are you getting my point? That is what you have to remember. Is that clear? Is that clear? Done. So we have the insects, we have the arachnids, we have the aquatic arthropods, right? Book gills are there, limulus or the king crab is there. Any doubts here, guys? Any doubts here? Then next to it, right, you will talk about the phylum mollusca. So in the case of mollusca, the second mollusca is what? Mollusca is the second largest phylum. Mollusca is what? It is the second largest phylum of animal kingdom. Are you getting my point? So in the case of mollusca, you will talk about Ctenidia, right? You will talk about Ctenidia, which is for the breathing as well as for the excretion. Is that clear? So here the Ctenidia, they are, you can consider them like, you know, some feather like gills. So here this is again the important one. So Ctenidia, it is meant for respiration as well as excretion. Are you getting my point? Respiration as well as excretion. So that's what you have to remember. Mollusca, right? Ctenidia is plural. Ctenidium is going to be what? Ctenidium is singular. Is that clear? Is that clear? Now, after that, what do we have? We have echinodermata. What do we have? We have echinoderms or echinodermata, starfish. So here we know that they don't have a proper respiratory organ. Rather, what will you see? In their case, we know that water vascular system, ambulacral system is present, right? Otherwise, other than that, what do we see? That body surface, you know, body surface here will have what? Body surface will have dermal branchiae, right? They have 2 feet in all, right? So 2 feet again. When we will discuss the animal kingdom, I'll teach you what water vascular system is, right? Then I will tell you. So what do we see here? So in the echinoderms, obviously here also what are we going to see? That body surface, right? Body surface, that is basically the dermal branchiae. They are present, right? Body, through body surface, this exchange will be called dermal branchiae in most genital bursae and tube feet. So this is what we are going to see. Please take a note. This is what we are going to see. Is that clear? Is that clear? Sure. So please take a note here. Quick. Yes, please. Done? Okay. Sure. Yes, beta, in the जैसे mollusca में different examples are there. You talk about the pila, there you see the pulmonary sac. So these are the, like, in general, I'm telling you the things here. Are you getting my point? So here we are reading the things in general. Okay. So after that, if I talk about what? If I talk about the chordates, if I talk about the fishes, we know it very well. They will have the gills. Okay. In the case of amphibians, right? Amphibian, you know that frog is the part of your syllabus. So you know that tadpole, which is the larva of the frog, right? You know that in that case you will see what? You will see the gills. And in the case of adult frog, what you people will see? You will see that skin is used for the exchange of gases. It can be used. The lungs are there. And even here, we talk about the buccopharyngeal cavity. Are you getting my point? We also talk about the buccopharyngeal cavity. Am I clear here? Right? Am I clear here? And even in the reptiles, in birds, right? In mammals, again, we'll talk about the lungs. Am I clear? Yes, please. Am I clear? So students, when we use the skin, when skin is the respiratory organ, when what is the respiratory organ? When skin is the respiratory organ, then the word used is cutaneous respiration. What's the word that we use? It is the cutaneous respiration. If lungs are used as the respiratory organ, then it is going to be pulmonary respiration. So you should be aware of these words. Then when gills are used for the respiration, so then it is the bronchial respiration, right? And when it is the buccopharyngeal cavity, so it is also named as buccopharyngeal respiration. Is that clear? Right? Is that clear? So this part you really need to know. So tell me if there is any doubt, then we will read it from the NCERT. Quick. Yes, everyone, quick. Any doubts? So write down and quickly revise it. Done. And here I didn't mention, I forgot to mention hemichordates. So in hemichordates, the exchange will be through, they have pharyngeal gill slits. I hope you remember the examples like belenoglossus, right? So that's what you have to remember. Am I clear? Am I clear? So this is about the respiratory organs and the different organisms. So let's revise it from the NCERT, right, beta? So beta, rather than say, ma'am, game of NEET क्यों बन किया बेटा? How does it matter? Under which series? What are we teaching? मतलब how does it matter? I'm teaching here. I'm completing a chapter. I think that's more important than any series name or something. Okay, so guys, please focus here, please pay attention. I don't want any such comment, right? Just learn the things here. So see, we'll cover the NCERT here in the lecture itself. I'll explain the topic, then we will start with the NCERT. So you should keep your NCERT with you, pen and pencil with you. So rather than keep a highlighter. So mechanism of breathing vary among different groups, and I told you it depends on what? It mainly depends on, highlight this part only, it mainly depends on their habitats and level of organization. So this is again a previous year question. Okay, so it depends on what? On the habitat as well as the level of organization. Now the next thing is lower invertebrates like sponges, poriferans, coelentrates, the example is hydra, flatworms like platyhelminths. So the exchange of gases is by simple diffusion over their entire body surface. We have completed that. Then earthworm, which is Annelida, they use their moist cuticle. Separately, I have mentioned it. Insects belong to phylum Arthropoda. They have the tracheal system, network of tubes. Okay, then they have special vascularized structure called gills, which are used by aquatic arthropods. I told you about the book gills as well, and mollusca, where vascularized bags called lungs are used by the terrestrial form, and you know that they also have the ctenidia. So this is what you have to highlight. Now, when it comes to the vertebrates, in the case of vertebrates, we are also the vertebrates. We have the vertebral column. Fishes, they use gills. Reptile, birds, mammals, they respire through lungs. Amphibian like frog, which is the part of syllabus as well. Moist skin is there. Right, mammals have well-developed respiratory system. So you know the details here. So this part here is very important. Please highlight this. So this is important MCQ. Like everything is important. This is very, very, very important. So on what basis it depends? On the habitat, on the level of organization. Any doubts? Any doubts? So next we have to start the human respiratory system. But before that, see, we'll discuss few terms here. Firstly, I'll tell you about the external respiration. Right? See, in the case of humans, we all are well aware that we have the pair of lungs. Yes or no? I believe you all know that. Yes? Yes? I believe you know that. Yes, please. Yes? So in the case of external respiration, so basically you know that in the case of humans, I'll discuss it in context of humans. Humans may, what do we have? We have lungs. We have lungs as what? As respiratory organ. Pair of lungs we have. Now, when you talk about the external respiration, here I'm trying to explain you a few terms. External respiration means when there is the exchange between environment. It is the exchange in between environment and organism. For humans, I'll specify here the blood, right? So when there is an exchange of gases, right? When there is an exchange of gases between the environment and the blood, in the case of humans, obviously you are talking about the blood, or in general, you can say when the exchange of gases in between the organism and its environment, right? That is going to be the external respiration. So in humans, we know it very well. It is the alveoli. Right, it is the alveoli which is the primary site for the exchange of gases. So here, this is a blood capillary. So that is how the exchange will occur, isn't it? That is how the exchange will occur, isn't it? So here, this is the external respiration. What's that? This is the external respiration. Now, we know it very well that this blood will carry the gases. It will carry it to the tissue. Yes, tissue is what? Group of cells having common origin. May or may not have similar structure and function, but having common origin. This is what tissue is. Now here also, right, here also you are exchanging the gases. This is basically the internal respiration. So when it is the internal respiration in humans, right, when there is the exchanges in between, yes please, when exchange is in between blood and tissue. When the exchange is in between blood and tissue. Is that clear? So this is going to be the internal respiration. So then comes what? Then comes the cellular respiration. So in the case of cellular respiration, what do you have? So here, when a cell it is using oxygen, it is oxidizing this glucose, then in multiple steps energy is releasing by using enzymes. We are trapping that energy to make ATP. That will come under the cellular respiration. Is that clear? That will come under what? That will come under the cellular respiration. So here you people can see, again clarifying NCERT, that utilization of oxygen by the cells, yes or no? Utilization of oxygen by the cells for catabolic reactions, right, for catabolic reaction, and the resultant is the release of CO2. This is what the cellular respiration is. So here you are talking about the breakdown of glucose in multiple steps by using enzymes and then the release of CO2. So this will come under the cellular respiration. I hope this term is clear. Now the next thing that we have to understand is indirect respiration and direct respiration. So you should know this. You should know these terms. These are important. So you should, literally you should know these terms. Firstly, direct respiration and then it is indirect respiration. So we have direct respiration and we have indirect respiration. That's what we need to discuss first. Am I clear? So anybody having idea of it? That what exactly is the direct respiration and what is the meaning of indirect respiration? Yes, please. Yes, everybody. Go for it. Explain it to me. Yes. What is the meaning of direct respiration and what is the meaning of indirect respiration? Anyone? Anybody? See, it indirect, as the name is saying, it doesn't involve, like here the exchange is not through any special respiratory organ and the blood is not involved there. And in the case of indirect respiration, you have the, right, you have the special respiratory organ here and moreover, right, you have the special respiratory organ here plus the blood is also involved. Are you getting my point? So we are talking about the direct respiration here. So we are saying it doesn't involve special respiratory organ or the blood. Is that clear? It doesn't involve special respiratory organ or blood. And obviously here we will talk about what? Special respiratory organs are involved. Special respiratory organs are involved and blood is used for transport of gases. Are you getting my point? So blood is used for what? For transport of gases. Is that clear? So blood is also used for the transport of gases. Is that clear? Yes, please. Right? So here, exchange of gases is through general body surface, by simple diffusion. By how? By simple diffusion. But here also, right, diffusion will occur indeed, but there will be the involvement of the special respiratory structure. Is that clear? There will be the involvement of the special respiratory structure. So these structures may be, right, so which structures do we have? Obviously, you know that gills, lungs, right, gills, lungs, etc. Right? So this is what we have here, even the buccal cavity. Okay, so if I have to quote the example, you have porifera, cnidaria. So it is occurring through general body surface, isn't it? Isn't it? Already told you about that. And moreover, one more thing. In the insects also, the respiration is the direct respiration, right? Respiration is the direct respiration. Do you know why? Yes, please. Do you know why? Anybody? Right? Here you know cutaneous, pulmonary, right, buccopharyngeal, and even the pulmonary respiration, we are aware of it, right? Bronchial respiration, we are aware of it. I hope here you know that we have cutaneous respiration where the skin is used, okay, this bronchial respiration, gills are involved, buccopharyngeal respiration is also there. So obviously buccopharyngeal cavity is involved there, and pulmonary respiration is there. So these are the special respiratory organs which are involved. And now here I'm saying that insects, this is important, right? That insects here also, they use the, they will also show the direct respiration. Please tell me, I'm just looking for the answer. Anybody? Yes, please. Why is it so? Why insects, they have the tracheal system, but why do we say that insects, they show us this, right? Why do we say that insects, they are showing us what? They are showing us the direct respiration. Yes, please. Anybody? Why? See, in the case of insects, we know that there are the tubes, right? Like spiracles are there, openings are there through which air will enter, and then further, tubes are there, and finally it reaches the tissue. So here we do not talk about the blood, right? It's just like it's a kind of, passage through where the air is coming, but there is no blood here. No blood is involved there. And then by simple diffusion, here also the exchange of gases will occur. You don't talk about the blood here. Blood is not involved here. Tracheal system, the tubes are just like, a kind of path through which the air will enter, and finally air will directly get exchanged here. So that is how it works. So any doubts? Yes, any doubts? Any doubts here? So these are the terms here. So you should know what external respiration is. You should know what internal respiration is. You should know what cellular respiration is, and what is the difference in the direct and the indirect respiration. I hope this part is clear. Yes, please. I hope this part is clear. Any doubts? Any doubts? Okay, so now let's talk about the humans. So where we know that, right, where we know it very well, that in the case of humans, right, what do you have? We'll simplify it. We know that pair of lungs. So let's talk about human respiratory system. Okay, let's discuss that. Okay, so in the case of humans, yes, please. So we are going to start with nostrils. We are going to start with what? We are going to start with the nostrils. So before going forward, right, what will we do? We are going to talk about this respiratory passage, then we will see what is present inside. So any doubt so far? Yes, any doubt so far? Hanji, quick. Some Darshan Karthik, ma'am is not replying to my doubt. But first of all, learn the spelling of replying, right? And you are here to spam, not to study, not to ask any question, and literally work on your spellings first. Okay? So those who are here for the study, very good. Those who are not, please don't waste your time. Don't waste my time. If you want to study, most welcome. If you don't want to study, I don't have any problem. Trust me. I don't have any issue. I don't have any problem. Okay? So let's get started. So as I said, first, what are we going to talk about? We are going to talk about the, right? We are going to talk about what? We are going to talk about the complete passage. So obviously, you know that we have the pair of lungs. So how are we going to start? External nares, right? Or external nostrils, nasal chamber, internal nostrils, then pharynx, larynx, trachea, primary bronchi. I hope you are aware of these terms, right? So this is what you all can see, right? I hope it is clearly visible to all of you. External nostril, which we also call as external nares, right? Then this vestibular passage is there, nasal chamber, internal nares or internal nostrils, whatever you want to call. Then comes the pharynx. So pharynx will have three parts here. Firstly, I'll start with it and then we'll go ahead. So pharynx having three structures. So for pharynx, you have to remember the trick knoll. What you have to remember? You have to remember the trick knoll. It means nasopharynx, then you have oropharynx, then you have laryngopharynx. Are you getting my point? Nasopharynx, oropharynx, and what else do you have? You have laryngopharynx. Naso, oro, laryngo. Naso, oro, laryngo. That is what you have to remember. Okay, that is what you have to remember. Okay, so external nostril nares or nostrils, vestibular passage, or you can simply say nasal chamber, internal nares, nasopharynx is there, then here oro and laryngopharynx, then this is the opening of the larynx, glottis, you have the larynx, then after larynx we have the trachea, primary bronchi, I hope you remember this. This is a windpipe, trachea, then it will bifurcate, you will have primary bronchi, then it will further bifurcate, right? We'll discuss it in detail. Right now, I'm just showing you this passage. So, secondary bronchi is there, tertiary bronchi is there, terminal bronchi is there, lobular bronchioles are there. Lobular bronchioles are there, and then there is the terminal bronchiole. So, up to this part, it is the conducting zone. Up to this part, which is conducting my air, right, to the area where the exchange of gases will occur, right? So we are going to talk about the human respiratory system. We know that we have the pair of lungs. So when we talk about the complete passage, how are we starting? We have the external nares, nasal chamber, internal nares, then after that pharynx is there, which is having three parts, naso, oro, laryngo, naso, oro, laryngo, then nasopharynx, then oropharynx, then laryngopharynx, then comes the larynx. Opening of larynx is glottis, larynx, trachea, primary bronchi, secondary bronchi, tertiary bronchi, terminal bronchi, then comes the terminal bronchi, then comes the lobular bronchiole, respiratory terminal bronchiole. So this is the part, right? This is the part where what are you talking? You are talking about the conducting zone. This is the part which will carry the air. This is the part which will bring it to the body temperature. This is the part where, you know, the humidification of air will occur. And then finally, respiratory bronchiole, alveolar duct, alveoli. This is the area where actually the exchange of gases will occur. So firstly, you have to understand this. Are you getting my point? You have to understand this, how the air is entering in this pair here, right? See, look at the diagram here. Okay, so we will simplify, we will clarify each and everything. So this is the complete passage. Let's have one more round of revision here. So how we started, external nares or nostrils, right? Then we are talking about the vestibular passage, nasal chamber, internal nares, then nasopharynx, oropharynx, laryngopharynx, then glottis, the opening of larynx, then larynx is there, then trachea. So trachea is the windpipe, right? Then primary bronchi, secondary bronchi, tertiary bronchi, terminal bronchi, and then finally lobular bronchiole will be there, terminal bronchiole will be there, then comes the respiratory bronchiole, alveolar duct and alveoli. So this is the exchange part, isn't it? Isn't it? This is the exchange part. So that is how, right? This is what you have to keep in your mind. Understood? Understood? So when you talk about the nostrils, so you know that we have the pair, right? So we have dirhinous condition because we have the pair, isn't it? What do we have here? So we have the pair here, right? So it is the external, a pair of external nares, and so basically a pair of external nostrils that are present above upper lip. Any doubts here? That are present above upper lip. So dirhinous condition. Which condition do we have here? Dirhinous condition is there. Am I clear? So we have the pair of external nostril, dirhinous condition. So where is it present? Right, these nostrils are present above the upper lip. Is that clear? Is that clear? Sure? Okay. Then it leads to nasal chamber through nasal passage, right? Yes, write down. Done. Write down. Yes, please. So now you know that, right? In our nose, right? In our nose, can you see that? We have a cartilage that is separating this nasal chamber, right? We know it very well that there is a cartilage here. Yes or no? There is a cartilage here. So this cartilage, so this is actually the hyaline cartilage, right? Hyaline cartilage is present here. You should know it. You know that we study that in structural organization in animals. So this is the, right? So hyaline cartilage, please write down, divides this nasal chamber into two. Am I clear? Am I clear? So it is the hyaline cartilage, right, students? Yes or no? It is the hyaline cartilage which will do what? It is basically dividing it into two chambers. So it is also the nasal septum. Are you getting my point? It is the nasal septum. Are you getting my point? Yes, please. Write down. Okay, so this is the, what is it? This is the nasal septum made up of what? Made up of hyaline cartilage. Are you getting my point? So students, these nasal chambers, right? These two nasal chambers, how will they form as two? Right? Because there is a partition. This is the nasal septum made up of hyaline cartilage, and the interior part of this chamber is basically the nasal vestibule. So nasal chamber is vascular. It is having the blood supply. It is having hair. It is having the mucus glands. Are you getting my point? So these here, these mucus glands, they will trap the impurities here. Are you getting my point? They will trap the impurities here. Understood? Understood? Done? Understood? So please write down. So nasal septum is the partition dividing the nasal cavity into two chambers. So nasal chamber is vascular, have mucus gland and have here, right? So you know that it will trap the fine particle. Mucus gland will secrete mucus for trapping them. Understood? Yes, please. Understood? So if there is any doubt, do let me know. Done? Okay. So you know their function. So here is there to trap dust particles, or if there is any impurities, right? Mucus gland secrete mucus to trap pathogens. So that's how it works. So that is not required here, so we'll not write it, but yeah. So as I said, interior part of the nasal chamber is basically the nasal vestibule, and posterior part is basically having the bones. It is having the three bones. Okay, and you know, these bones, they also form the outgrowth like structure. So when the air passes through it, right, because in the bones part also you will see some outgrowth, some spiral outgrowths. So obviously area is also increasing. So this thing is going to do what? It will increase, right? It will basically humidify the air. It is going to bring it to the body temperature. It will also humidify the air. So this is how everything is related. So I hope this part is clear to everybody. Yes or no? Yes or no? Okay. Sure? Done? So what I just said, that the posterior part of nasal chamber is having three bones. Posterior part of nasal chamber is having three bones, and these bones form some outgrowths that will make nasal chamber spiral and long. Okay, so it helps to bring air to body temperature and humidifies it. Number one. Moreover, so this part of nasal chamber is having olfactory epithelium. That's why you are able to smell the things as well. Is having olfactory epithelium in some part and pseudo-stratified ciliated columnar glandular epithelium in some. So I hope it's clear to you. So our nasal chamber, right, the epithelium here is olfactory, as well as some of the portion is olfactory, other is this pseudo-stratified ciliated columnar glandular epithelium. So let me talk about it. Pseudo means false, stratified means layered. So it looks layered, but it is actually single layered. Ciliated, it is having cilia over it. Columnar means cells are tall and cylinder. They have the cilia. They look like multi-layered, but actually it is single layered, right? Even these cells are secretory. So this is what we have to discuss. I'll use the same term, same short form while explaining the trachea as well. So you should know this. So firstly, take a note of this part, then I'll move to the next portion. Quick. Yes, please. Quick. Guys, quick. Any doubts? Done, beta? Noted? So let's move to the next part. So it is pseudo-stratified ciliated columnar epithelium. Understood? Understood? So the nasal chamber, let's be quick. So the nasal chamber opens into nasopharynx. I told you pharynx is having three parts, naso, oro and laryngo, but how? Through internal nostrils. Am I clear? How? Through internal nostrils or internal nares. Is that clear? Is that clear? So the pathway clear. So this is what you have to mark. So nasopharynx will open into the nasal chamber will open into the nasopharynx. Is that clear? Done? Yes, please. Now, as I said, pharynx is having three parts, right, students? I told you this thing. So nasopharynx is there, right, students? You have oropharynx. So again, I'm repeating this. So third one will be laryngopharynx, right? Third one will be laryngopharynx. Understood? So nasopharynx, oropharynx, laryngopharynx, that is what we have. Are you getting my point? Nasopharynx, oropharynx, laryngopharynx. So this is what we have. Understood? Done? Done? So any doubt here? Any doubt here? So naso, oro, laryngo. Naso, oro, laryngopharynx. These are the three things. Now see, nasopharynx through internal nares, it will receive air. Then comes the oropharynx. Now understand one part here. See, this is mouth and then the buccal cavity is there. We are eating it. That food will also pass to the pharynx. So pharynx is a common passage. If you will read the NCERT where the digestion and absorption, the old NCERT where the digestion and absorption chapter used to, the digestion and absorption chapter was there. So there they used to mention that pharynx is the common passage for the air as well as for the food. So if we talk about the pharynx, as I said, we are dividing it into three parts: naso, oro, laryngo. Naso, oro, laryngo. Repeat again: naso, oro, laryngo. Is that clear? Right? Is that clear? So it is going to be naso, oro, laryngo. Naso, oro, laryngo. These are the three things that we have. So nasopharynx will receive air. Now you are eating it. Food will pass. That is the oropharynx. Will have both food and air. This is common for both. Then comes obviously laryngo. It will further pass to what? Laryngopharynx. Are you getting my point? It will further pass to what? It will further pass to laryngopharynx. Now when you talk about the larynx, see, now understand one thing. This is our dorsal side. This one is our dorsal side, ventral side and the lateral side. I'm repeating this again. Dorsal side, ventral side, then comes the lateral side. So here, right, this is our ventral side. So here you will see, like, see, pharynx. It is going further, right? Now that's how you have to understand this part with the help of this diagram. So you are having your food, it is in the pharynx. This part here, this opening here is basically glottis, right? This opening here is what? It is the glottis. Glottis, and then here you have the cartilaginous box larynx, right? You have the cartilaginous box larynx, and then it will lead to the trachea, and just behind it, this is the ventral side, of course, I'm talking about. So just behind it, you'll see the esophagus, and I know you all know that it is the food pipe, and the opening of the esophagus is what? This is basically gullet. What's that? It is gullet. So opening of esophagus is gullet, but the opening of this trachea, of this larynx, is glottis. So when we eat food, it is always advised that we should not talk while eating. Why? Because food should ultimately pass here, right, by a glottis into esophagus. If food will enter here, obviously that will be problematic. So when we swallow the food, right, so during swallowing of food, there is a cartilaginous flap here, right? There is a cartilaginous flap, called epiglottis, covers the glottis. That's what you need to know, and this is obviously an important statement. Kindly write down, kindly mark it. Yes, please, quick. Write it down and mark it. Understood? Done. So it is a cartilaginous flap. It is a cartilaginous flap. Noted? Yes, everybody, noted? Done? So this is an important question. So this is important. It's a cartilaginous flap, means it is made up of cartilages. It's a kind of, leaf-like flap of elastic cartilage. That's what you can write down here. So it is what? Elastic cartilage, made up of elastic cartilage. So that is what you guys can write down. And now I'll show you this diagram of NCERT, because diagram based questions, they can come in paper. So you guys can see this is what they are trying to show, a triangular or the leaf like. So it is epiglottis. So it's a cartilaginous. In the book also, that's how you have to mark the things. So it is the cartilaginous flap. It is the cartilaginous flap. And which cartilage it is? It is the elastic cartilage. It is the elastic cartilage. Is that clear? Is that clear? Done. And then comes the larynx. So larynx is what? It is the voice box. Now let's talk about it. So larynx, voice box, a cartilaginous box, because it is made up of cartilages. So here you can see, then comes the trachea. And what these structures are here on the trachea, these are the C-shaped cartilaginous rings. Understood? So that's what we have to discuss now. Any doubts from the diagrams? Yes, please. Any doubts from the diagrams? Yes, please. Sure? So look at the diagram. Look at this diagram. So this is the ventral side, you know that. Dorsal side, right? And lips, okay, the food will enter. This is the oral cavity. Here you can see uvula. This is the pharynx part, the common for it. Now you guys can see this is epiglottis. Can you see that? This is epiglottis. This one is the larynx. So this is the ventral side. This side will have trachea. Behind it, esophagus is there, right? So esophagus opening is known as what? It is the gullet, G-U-L-L-E-T. And this epiglottis will cover it when you swallow the food, so that food should not enter here. Understood? So let's have a look. Just look at it. That's the hyoid bone, U-shaped bone that supports tongue. That supports what? That supports tongue. You can see that. So then it is the larynx. As I said, it is a cartilaginous box made up of cartilages. So it is having this one, thyroid cartilage, right, cricoid cartilage, tracheal cartilage. So this cartilage is the largest cartilage actually. Not written in NCERT, just for your knowledge I'm telling you this. So it is the largest cartilage. So look at the structure here. Then these are the tracheal cartilages, that C-shaped cartilaginous rings, right? So look at all the cartilages here. Look at this diagram. This is that epiglottis, that cartilaginous flap. And then here you guys can see this is the false vocal cord, right? This is the false vocal cord. Can you see that? This is ventricular fold, false vocal cord. So two vocal cords are also there, the true one and the false one. False one, obviously it is not producing any sound, but the true one will reach through this side. So the diagram part clear? Yes, everybody, the diagram part clear? Quick. Any doubts? So see this hyoid bone, already told you, supports tongue, attached to the thyroid cartilage by thyrohyoid membrane. So look at the diagram here. So I hope diagram part is done and dusted. So let's talk about the larynx, voice box. So you know that in the case of boys, when they hit the puberty, right, this larynx, it forms what? Adam's apple. Adam's apple. Do you know that? Adam's apple. right? So let's talk about it. Be ready. Okay, let's talk about it. Just a minute, guys. So larynx, cartilaginous box that helps in sound production, right? So it's a cartilaginous box that helps in sound production. Are you getting my point? Cartilaginous box that helps in what? That helps in sound production. So called voice box, present in between third to sixth cervical vertebrae and a little higher in women and children. Understood? So largest cartilage, I told you, that much detail is not required for your syllabus. So write it down. Make sure you write all the points required. Okay? Okay? Yes, please. Done? So next is the cavity of larynx contains two pairs of mucosal folds. Done, students? Number one is upper, or also known as vestibular fold. This one is actually the false vocal cords. What are they? False vocal cords. So they do not help in sound production. Are you getting my point? How many of you are aware of it? Yes, so upper or the vestibular fold, it doesn't help in sound production. Are you getting my point? It doesn't help in sound production. They just provide moisture to the true vocal cords. Okay, right? So they do not help in sound production but provide moisture to true vocal cords. Am I clear? That's what you have to remember. So second one is the lower fold, which are basically the true vocal cords, right? Helps in phonation. These are actually the one which will helps in phonation. So phonation means sound production. Done? So phonation means sound production. So that's what you have to remember. Any doubt? Yes, please. Any doubt? Done? Understood? So upper fold is there and then comes the lower fold. So it is the true vocal cord that helps in the phonation / sound production. Understood? So this is about it. And here I would like to mention one more thing, that in birds, you know now the kind of sound they produce, syrinx help in sound production. Done? Syrinx helps in sound production. They do have larynx, but larynx is not playing any role in the sound production. It is the syrinx in the birds. Okay, it is the syrinx in the birds. But in birds, yes, larynx is also present, but playing no role in the sound production. It is the syrinx. Okay, it is the syrinx. So this part is clear, guys. This part is clear. So anybody having any doubt? Anybody having any doubt? Yes, please. Arjun, wherever you want to study, please go for it. please go for it. Right, I am here to deliver the quality content. I'm not bound to do this, that I have to finish the chapter in 3 hours or in 2 hours or in this and that hours. I'll clear all the concepts along with NCERT. I'll provide you the extra information which is required to clear this exam. So you want to study here, do it, otherwise leave it. Right, bye bye, tada. So up to this part all clear. So larynx is the voice box or the sound box, and in the males, when they attain puberty, you know that, right? It will, we call it as what? Like here we see it as an outgrowth now in the case of males. So we call it as Adam's apple. So anything, right? Yes, anything else? Yes, please. Anything else? Okay, so do mark it, quick. Do mark it, everybody, quick. Done. So, vocal cords, they are larger and thicker. They are larger and thicker in men, and obviously smaller and thinner in women. So told you this thing already, that what? Told you this thing already, that basically the sound production, it depends upon this thickness and thinness of the vocal cords as well, right? So that is why in the male and the female the sound is of different pitch, because you can see vocal cords are large and thicker in the men, and in the women it is going to be smaller and thinner. So these are minor details that I believe you should know about your system. So I hope up to this part everything is clear. Okay, chaliye. So next part. So before starting the next portion, again we will simplify it from the NCERT as well. So let's get started. So again, come back to the NCERT part. So here you can see pair of external nostrils opening out above the upper lip. Done. Leads to nasal chamber. How? Through nasal passage. Again, if you will solve the PYQs, you'll find this question, right? And it's a very simple one. Then nasal chamber, it opens into the pharynx. The nasal chamber, where will it open? Into the pharynx. So here you can see it is clearly written that it is basically the common passage, and I told you it is having 3 parts. The trick for that is knoll, nasopharynx, oropharynx, laryngopharynx. So literally this oropharynx is common for both food and air firstly. So then again the next part is that pharynx opens through the larynx into the trachea, but you know that here you will see the larynx, that pharynx opens through the larynx into the trachea. So larynx is the voice box, just now we discussed. So it is the cartilaginous box, help in sound production, right? That's why the sound box. So during swallowing, glottis can be covered by a thin elastic cartilaginous flap called epiglottis. So if I have to frame the question from this part, instead of elastic, I can mention hyaline, I can mention something else as well. So accordingly you have to pay attention. Then trachea. Now next part, obviously we'll talk about the trachea, which is a straight tube extending up to the mid-thoracic cavity, divides at the level of 5th thoracic vertebrae into right and left primary bronchi. So that is the reason, right? That is the reason that I told you about the respiratory passage first. Okay, so now let's simplify this portion. I believe larynx is done. So now here let's talk about the trachea part. So what we just discussed, that it will divide into, it will bifurcate into primary and secondary bronchi at the level of 5th, yes, at the level of 5th dash vertebrae. So 5th dash vertebrae, 5th thoracic vertebrae or 5th cervical vertebrae, what should be there? I can see a student is mentioning the answer here, but yes, what should be there? 5th thoracic vertebrae or something else? Quick. Guys, quick, answer it. 5th thoracic vertebrae or 5th cervical vertebrae. So these kind of things, this is how the question can be framed in final paper, and that's how you have to study the NCERT, the topics, the concept, that you should know this thing. So what are we talking? Exactly. Okay, let's go for it. Right? So thoracic, sorry, trachea, it's the windpipe. It's the windpipe through which air will pass. Now you tell me, in the case of trachea, no doubt, firstly, you know that this is our dorsal side, ventral side, hai na? This trachea, it is placed here on the ventral side. Now, when we talk about the vertebral column, which is placed dorsally, right? Vertebral column, which is, where is it present? It is dorsally placed now. So here we have CTLSC. What do we have here? We have CTLSC. What do we have here, guys? We have CTLSC. So cervical is there, thoracic is there, right? Lumbar is there. Cervical is there, thoracic is there, lumbar is there. Then comes the sacral coccygis, या coccygeal region. That's what we study. So what is the formula in our case, right? It is C7, T12, L5, L5 fused, which will result in the formation of one vertebrae. Then C5 fused, C554, sorry. C is 4 here. Again, it is fused, will result in 1. So we have 26 vertebrae, 7 plus 12 plus 5th plus 1 and 1. Right? So before fusion, it is going to be what? Right? You can count it. It is 33, isn't it? Right? And after fusion, it is going to be 26. So when you talk about the trachea, so when it is like trachea, we have 12 thoracic vertebrae. Now, if I write it like this, just to show you, so basically what am I trying to tell you? That at the level of 5th thoracic vertebrae, see, so vertebral column is dorsally placed, this ventrally we have this trachea. So at the level of 5th thoracic vertebrae, it will bifurcate into 2. That's what we are talking. Are you getting my point? So at the level of 5th thoracic vertebrae, right, it will bifurcate into 2. Vertebrae, it bifurcates into primary bronchi. Am I clear? Sure. And we know that trachea and further parts, we'll discuss that also. Here we see what? We see these C-shaped cartilaginous rings. C-shaped cartilaginous rings. Please tell me why. Why they are C-shaped? Why are they not? They are not covering it completely. Yes, please. Just say, let's suppose if you are wearing a ring, right? That will cover all the sides of your finger, no? But here, when you are talking about it, it is C-shaped. So I really want to know the answer. I want to know the reason. Yes, please. I really want to know the reason here. I really want to know the answer here. Why is it so? Please tell me quickly. Why is it so? Yes, everyone. Why is it so? Why it is C-shaped? Why it is not completely covering the trachea? Answer it to me. I'm reading the charts. Quick. Quick. Anybody? Yes, please. Anybody? They provide the support only to trachea because presence of esophagus to expand. Very good. See, basically, let's suppose I told you already this thing. Just say, let's suppose the diagram, obviously, this is just to explain you, not the appropriate one. Just say, let's say this is the dorsal side. This is the ventral side. Now, this is the trachea, and just behind trachea, what do you see? You see the esophagus. Okay? This is the trachea, and just behind trachea, you see esophagus. Now, we are saying that trachea is having C-shaped, obviously, these are C-shaped cartilaginous rings, right? These are C-shaped cartilaginous rings. So, this empty part is towards, that incomplete part or the empty part is towards, is posteriorly. This incomplete part is present posteriorly. Are you getting my point? Posteriorly. So what does it mean posteriorly? Matlab towards esophagus side, right? Towards esophagus side, you are going to find it. Where are you going to find it? You are going to find it towards the esophagus side. So I am talking about the trachea here. I am saying that it is having the C-shaped cartilaginous rings, and I'm telling you clearly this is going to be, this is the hyaline cartilage. So that C-shaped part, that incomplete part is towards the posterior side, means the side of the trachea which is facing your esophagus. Now, first of all, right, why it is required? Why these C-shaped rings are required? Because they are going to provide the rigidity to the trachea. You know that we inhale through the simple diffusion, isn't it? There's a first reason here is rigidity, to prevent tracheal collapse. Now, how is it possible? Obviously, it is providing the rigidity. You know that when we inhale, inside there is the negative intrapulmonary pressure, right? We'll discuss it in the next topic. So inside we have the negative intrapulmonary pressure, right? That is why air, basically diffusion is what? The gases, they will move from their higher concentration, like what in general, what is diffusion? The movement from high to low. So in gases, obviously wherever there is the low partial pressure, they will move from high to low. So during inhalation in the lungs, the negative intrapulmonary pressure is there, right? So just to prevent this trachea to get, just to make sure the trachea should not collapse, these cartilaginous rings are there. They are protecting it. They are preventing the collapsing. Secondly, just here we have the esophagus. So when we eat the food, esophagus will also expand. So if here will be the tracheal rings, that will, you know, prevent that, that will obstruct that food passage, right? So now it is incompletely covered. So whenever we'll swallow the food, esophagus will also expand. So that incomplete side, that incomplete tracheal rings, they will not provide any obstruction here. So that's why they are incomplete. Is that clear? Is that clear? So both the reasons are there, and I hope you know about them. Understood? Done? Done? So next thing in simple words you can write as flexibility, right? So it is providing flexibility to the esophagus. Okay? So it permits esophagus dilation during swallowing of food, which is also known as deglutition. Swallowing. Done? So that's why they are incomplete. Am I clear? That's why they are what? They are incomplete. Any doubts? Any doubts? Okay. Sure? So this trachea, it is also lined by pseudo-stratified ciliated columnar glandular epithelium. Told you already about that. Look at this passage part, then you will understand it in a better way, right? Just say here, we have this part, right? We have this part. So from trachea, so trachea is basically this pseudo-stratified ciliated columnar glandular epithelium, right? Right, so yes, it is also a PYQ. So you can see till basically your tertiary bronchi, you are going to find this epithelium. So it's pseudo stratified, means it looks like multi-layered, but actually it is single layered, ciliated cells, columnar cells having cilia, plus it is also glandular. So it will release mucus that can trap if there is any, you know, pathogen or any impurity enters in our body. Okay, so columnar cells, repeating myself again, tall and cylinder, and nucleus is present at the base. So it is tall and cylinder, nucleus is at the base, and these are small, small, small. This is the cilia. So the beating of cilia will help in, if there is anything that enters in our trachea, right, this will be trapped in the mucus, then the movement of this cilia will help to throw it out, So this is how it works. So this part is clear. This part is clear. Yes, everybody, this part is clear. So from this trachea, right, everybody, so from this trachea, you will see that up to the thin terminal bronchioles, right, repeated divisions are there, and till initial bronchioles, till what? Till initial bronchioles, you people will see that C-shaped cartilaginous rings are present. Am I clear? So C-shaped cartilaginous rings are present up to this point, and where this trachea is bifurcating into primary bronchi, so you call this point as carina, right? Carina, where it is bifurcating. Now again come back to the NCERT part so that we can highlight it as well. So here you people can see trachea is a straight tube extending up to mid-thoracic cavity. It will divide at the level of 5th thoracic vertebrae. Explained it to you, right? So this is obviously very important. So it is left and the right primary bronchi. Then bronchi will undergo the repeated division to form the secondary and tertiary bronchi and the bronchioles, ending up in very thin terminal bronchiole. So obviously this will be in the exchange zone. So trachea, primary, secondary. So this is what I just discussed and marked in this flowchart there. So look at this. Look at this. Trachea, primary, secondary, tertiary bronchi, and then so this is the thing. They are supported by incomplete cartilaginous rings. So yes, this question can be asked in the paper. Do underline it. Okay, do underline it. Done. So each terminal bronchiole will then give rise to very thin irregular walled and vascularized bag-like structure alveoli. So branching network of bronchi, bronchiole and alveoli, it is basically making the lungs, right? So we have the pair of lungs covered by pleura. So next, this is the part that we have to discuss. And here I told you that the part where, this trachea is bifurcating into those two, we are calling it as carina. So that's what we have to discuss. I hope the diagram and everything is crystal clear to all of you. Understood? Understood? Sure? Sure? Any doubts here? Let's talk about the lungs and then we will talk about the alveoli. But before that, see, again from the NCERT, look at this, guys. Again, we have already covered this part, that the part which is starting with the external nostril, from external nostril up to terminal bronchioles. Pay attention. Already added the flowchart there, right? Already added the flowchart there. So from this external nostrils, yes please, from this external nostril to the terminal bronchiole, this is the conducting part. What is it? Conducting part. But alveoli and their ducts, they form the respiratory or the exchange part or the respiratory system. So conducting part is actually what? Conducting part will transport this air to the alveoli, which is the primary site for the exchange of gases. It will clear it from the foreign particle. It will humidify this air and bring it to the body temperature. And exchange site is the place where there is the actual exchange of gases. Are you getting my point? Where there is the actual exchange of gases. We'll show you the diagrams also, but before that, look at this part. So here you guys can see, please have a look. See, so respiratory, bronchiole, alveolar duct, alveoli. So this is actually the exchange site. So from this external nostril to this terminal bronchiole. This is just the conducting zone. So conducting zone told you already. Yes, please just discuss it with me once. So what will that conducting zone do? Carry air to exchange zone. Humidifies it. Bring that air to body temperature and trap FOH, foreign particles. So that's the role of your conducting zone. Am I clear? That's the role of your conducting zone. So let's talk about the next part, that is the pair of lungs. So here our lungs are present, right? First discuss it, then we'll go ahead. So lungs, they are in the thoracic chamber. Okay, now lungs, very delicate organs, right? Pair of lungs we have the right lung, we have the left lung. The space present in between these two, it is known as mediastinum, where you will see the heart, the thymus, the part of esophagus will move through it, etc, etc. Now when you are talking about the lungs, they are obviously covered with the membranes, which we are calling as pleural membranes. We have outer pleural membrane, we have inner pleural membrane, we have the space in between pleural cavity, which will have the pleural fluid, which will obviously, that will lubricate that part and will reduce that friction thing. Now when you're talking about the lungs, because they are very delicate, right? Now understand this thing, dorsally, right? You already know we have the vertebral column, we have the vertebrae. To be more precise, we have the thoracic vertebrae. Now when you see ventrally, here we have the sternum, the breastbone, and then ribs are also there. Laterally and this side, ribs are also there, pair of ribs. So they are protecting the lungs, right? They are protecting the lungs. And meanwhile, this side, lower side will have the diaphragm, right? Lower side will have what? Lower side will have the diaphragm. So when you talk about the lungs, they are present in this airtight chamber. Okay, where these lungs are present, they are present in this airtight chamber. Just have a look of this structure, right? They are present in the airtight chamber. You will see this is the sternum, the breastbone. You can see the ribs here, pair of ribs here. So this side is having the diaphragm, right? Diaphragm. So this diaphragm is dividing this thoracic and abdominal. This cavity, this thoracic and abdominal cavity is divided by it. Okay, present in between this. So this is something which is unique to the mammals, diaphragm. Are you getting my point? So it is an airtight chamber. It is an airtight chamber where your lungs are present, right? Where your lungs are present. And do you know that lungs, they cannot, like within the lungs, obviously lungs, within the lungs will be the pulmonary cavity. So they cannot control their volume. So lungs are placed in this airtight chamber in such a way that whenever there is any change in the volume of the thoracic chamber, that will reflect in the volume of the pulmonary cavity as well. So that's the part that we have to discuss now. Here you guys can see the structure as well. Look at this side, it is your right lung, right? This is your left lung. We know that heart is placed here, a little bit pushed towards the left side. That's why this left lung is having this cardiac notch also, right? This left lung is having cardiac notch. So cardiac notch is there in the left lung. Okay, this is in the left lung, not in the right lung. So right lung is going to be, you know, shorter, broader. This is longer, narrower. Right heart, you know that it is in the space present in between the lungs. So we are calling it as the mediastinum. So lungs are nothing but the soft, spongy, pinkish, elastic structure. So let's discuss about the lungs. Ready? Ready? Sure? Sure? Okay, so lungs, endodermal in origin, means made up of endodermis, placed in thoracic cavity, which is anatomically an airtight chamber. What's that? Which is anatomically an airtight chamber. Am I clear? Am I clear? And they are present on the either side of the heart. Yes or no? Yes or no? Both the sides of heart. Now, lungs are covered by double layer of pleural membrane. We also call it as pleuron. Are you getting my point? So it is also covered by double layer of pleural membrane. Are you getting my point? So let's see. I'll name it here. Just say this is outer or parietal pleural membrane. This one is your inner or visceral pleural membrane or pleuron. That's why I told you it is the double layer. Now you people can see the space in between. We call it as pleural cavity, right? Having pleural fluid. Is that clear? Having pleural fluid. Any doubt here? Yes, please. So outer one is parietal, inner one is visceral. Understood? Inner one is visceral. Is that clear? Yes. So it reduces friction on lung surface and acts like a shock absorber. Am I clear? And acts like a shock absorber. Is that clear? Is that clear? So that's what you have to remember. So outer parietal pleural membrane, inner visceral pleural membrane, and this outer one is obviously close to what? Where is it present? Thoracic cavity, right, to that anatomical air chamber. So in between we have the pleural cavity, which is fully loaded with the pleural fluid, and as I told you, it will reduce the friction on the lung surface and will act like a shock absorber. Is that clear? Is that clear? Done? Sure? Sure? Any doubts? So other than that, what else you need to know? So the next part is that the space between two lungs, already explained you this, is called mediastinum. This is the mediastinal space. It contains heart, obviously part of esophagus. It is having your thymus. It is having your thoracic duct, even the pleural membranes. I told you how, etc. are present here, are present in it, right students? They are present in it. Here, hilus is present. So what is this hilus? From where blood vessels, lymphatic vessels, lymphatic vessels, nerves, etc. enter or leave. Done? So hilus is also present. So this is the mediastinum. Just now I explained you this, so you can see this. देखो, lungs are covered. Lungs are covered with the pleural membrane. Obviously, when lungs are covered by the pleural membranes, right? So obviously, if there is the outer pleural membrane that is in close association with the thoracic lining, inner one will be with the lungs. So they are completely covered. So obviously, this area in between is mediastinum. Here also we see the pleural membranes. We see the esophagus will also pass. We also see the heart will be there. So that's what you have to remember. Got it? That's what you have to remember. So this is so that's how it is present. And obviously, as I said, that it is covered with what? It is covered with lungs are in anatomical airtight chamber. So you should know this is actually the thoracic cage or the rib cage, the thoracic basket. So here you should know dorsally what is present, vertebral column. To be more precise, just a minute. Oh. I'm unable to move it, I think. So you people can see this here. So dorsally vertebral column, especially thoracic vertebrae, then ventrally sternum, which is also known as breastbone, laterally ribs, right? And lower side will have dome-shaped diaphragm. Am I clear? Dome-shaped diaphragm. So that's what you have to remember. Very important. Very important. So this is your thoracic cage or rib cage. Understood? Understood? Okay. Any doubts? Any doubts? Noted? So now after this I'll teach you a very important part, but tell me if there is any doubt. Done? Okay, so next is so lungs, they are soft, spongy, elastic and pinkish at birth. Do you know that? Do you know that? So when you talk about the right lung, already told you, we have the left lung. So right lung is basically shorter, broader, longer, narrower. No cardiac notch. I told you what cardiac notch is. Cardiac notch present. Have 3 lobes. Have 2 lobes. Do you know that? So basically when you talk about the right lung, you will see that there will be, two fissures that are going to divide it. right? But here in the left lung, there is only one fissure. So this one fissure will divide it into two lobes. Here there will be three lobes. You will have superior, middle, and then inferior one. Okay, and then inferior one. Got that? Right? So three lobes are there, superior, middle, and inferior. There you have only two, superior and inferior. Understood? Sure? Any doubts? Yes, please. Any doubts? So right and left lungs clear? Sure? Okay, so one more line I have to explain, but again, I'll discuss about the diaphragm first. So I told you already, it will divide the thoracic cavity and the abdominal cavity, right? It will divide thoracic and the abdominal cavity. Obviously, a true diaphragm is present in, it's a unique feature of mammals only. And students, it's a, basically it is what? Right, so diaphragm, it's very, very important. It's very important for the respiration. The next topic which we are going to discuss here is, so you should know about it. So it's a dome-shaped, you know that, dome-shaped structure, right? It's a dome-shaped structure, okay? Right, so it's a muscular and a fibrous septum. That's what you have to write. It's a muscular and fibrous septum, principal muscle for respiration. It is also having an opening, right? Also contain opening through which esophagus pass. Understood? So it will help in the respiration. We will see how. Done? Yes. Yes, Jessica. Okay, so it is having the opening through which, you know, your not just the esophagus, even the aorta, vena cavas, they pass. Inferior vena cava, of course. Done. So diaphragm done. So it is basically having the peripheral skeletal muscle, central tendinous sheath. So that's what you have to remember. So that's how it works. So you can say the true diaphragm, right? True diaphragm, where do we see? True diaphragm we will see in the case of mammals. Okay. True diaphragm, where do we see? We will see it in the case of mammals. Understood? Understood? Chalo. So now a very, very important line. This one. Again, I'm repeating it. This lungs. Obviously, the space in between the lungs, it is basically the pulmonary cavity. Lungs cannot control their volume. They cannot increase or decrease the volume of this pulmonary cavity. But, what is the important thing here, guys? What is the important thing here? Lungs cannot do that. Lungs cannot control that. But what is the important thing? The important thing is that lungs are placed in this anatomical air chamber in such a way that if there is change in the thoracic cavity's volume, that will reflect there. And that is something which is very important for the respiration. If we have to respire, we'll talk about the Boyle's law there, right? Then you will see that there should be a change in the volume, and then only the air will come and go. So please mark this, and if there is any doubt, do let me know. See, again I'm telling you, the thoracic chamber, you know that dorsally having vertebral column, to be more precise, thoracic vertebrae, ventrally sternum, laterally the ribs are there, lower side is having dome-shaped diaphragm. So, a very, very, very important line, the anatomical setup of lungs in the thorax is such that any change in the volume of thoracic cavity will be reflected in the lung cavity. Such an arrangement is very important to alter the pulmonary volume. So, I will add the things here. So, listen to me. I'll add the things here. So, listen to me. Right. See, just say if exam is tough, you know that the cutoff will go down. If one is increasing, another is decreasing, inversely proportional. Now, next thing, if you prepare well, you will have better result. If you will not prepare well, result will also not be good. Same as the case here. We know that outer pleural membrane is in close association with the thoracic cavity. Inner pleural membrane is in close association with the lungs. Now, this arrangement is in such a way, because lungs are not itself altering its volume. Then, if thoracic cavity, because they are attached like that, so if thoracic cavity, if it is increasing its volume, the lungs will also, the pulmonary volume will also increase, pulmonary cavity volume. If it decreases, then it will also decrease. So, that is something very important you have to keep in your mind. Understood? Understood, a very, very important part here. Done. So, look at the diagram, thoracic trachea, cervical trachea, larynx part, right? So, at the level of fifth thoracic vertebrae, it will bifurcate, right? So, primary bronchi, and then see, this is trying to show us, right? This is a wonderful diagram. Can you see that? This is the sternum, right? Manubrium and rest of the body, xiphoid process. Look at this. Look at this. That's how it is protecting it. This much portion, right? This thoracic trachea portion. See, that's why I told you thoracic trachea to be more precise. Now see, told you pseudo-stratified columnar ciliated epithelium. So it looks like multi-layered, but actually it is single-layered, right? This is the diagram, right? So this is carina. I told you the point from where it will bifurcate. The point from where it will bifurcate. So here they are showing you tracheal muscles, hyaline cartilage ring which is C-shaped. This is the lumen, and look at the ciliated epithelium here. Look at the mucosal glands are also present there, right? So mucus, mucociliary ciliary escalators, debris are there. Cilia will beat, it will, so that it will come back, and you know we can just throw that out. So goblet cell, which are the mucus cell, right? They are present. Cartilages, look at the cartilages there. So this is the diagram. So from this part you are getting this internal structure. From this part you can see this. So these are two different sections. You can just have a look. Okay. So I hope you liked it. I hope you liked it. Now we will talk about the alveoli, but before that just try to answer this question. A very easy one. Just try to answer this, then I will give you a break for 15 minutes. We'll have dinner and then we will continue it. Right? We will continue it. Chalo, quick. Quick. Answer this question. Hanji, what should be the answer? Exactly. So it is going to be the epiglottis. Chaliye next. 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 Again, very easy. Yes or no? Yes or no? So A with 3, you got your answer. Right? A with 3, you got your answer. Oh, acha. 1 second. Not A with 3, my bad. You're talking about the, yes, spider will show you what? Book gills or book lungs? Spider will show you what? Book gills or book lungs? Hanji, quick. Book lungs. So A should go with 2, my bad. So then B will go with 1. King crab. Limulus it is. We discussed it today only. So C will go with 1. Labiu will also go with 1. Chaliye, next. Lungs are enclosed in. Very easy. Lungs are enclosed in. Pleural membranes. ठीक है? Now we have to discuss the alveoli and the capacities. Chalo. So we'll have a break. Break for 15 minutes. So I'll give you a question. How can we figure out that if it is a stillbirth, that is baby born dead, or it's a murder? How can we figure it out? So this is a question for you, right? Thank you so much. So this is a question for you. So rest we will discuss after 15 minutes. So let's have a break for 15 minutes and then we will come back for the next topic. So Hi everyone, so let's start. So any answer for this question? Yes. Any answer for this question? Any answer? Anybody? How can you differentiate the stillbirth, that a baby born dead, or that baby is murdered? It's simple, like there are other ways also, but lung floating test can be done. So do you know about it? Yes, do you know about it? Lung floating test can be done. Just a minute, guys. So it is the lung floating test, also known as hydrostatic test. So what exactly is happening here? So if there is air in the lungs, right? If there is air in the lungs, then definitely the lungs are going to float. They will not sink, they will float. But if there is no air in the lungs, then lungs are going to sink. So that is how we can check. Because if it is a stillbirth, means there is no air in the lungs, so the lungs will do what? Yes, please. Lungs will do what? Because there is no air in the lungs. Yes, students? So if they sink, so means baby did not breathe at all, so there is a stillbirth. But if they float, then it is a murder. So this is what you can know. So now the next thing is the alveoli. We will talk about the alveoli and then we will talk about the capacities. So any doubt so far? Any doubt so far? Yes. So if you guys are having any doubt, do let me know. Quick. Quick. Yes, please. देखो बेटा, before starting the alveoli, because I believe you all know that it is the primary site for exchange of gases. It is the primary site for what? It is the primary site for exchange of gases, and we will talk about its epithelium. Simple squamous epithelium is there. You know that alveoli, we sometimes also use the word atrium or atria, but these two are the different things. There is a difference in that. Do not confuse this atria with the atria that we discuss in the heart, right? No need to, right? No need to confuse there. So when you talk about the alveoli, you know that they are the primary site for what? They are the primary site for the exchange of gases. Yes or no? Yes or no? Yes or no? So it is, we know that primary site, and this one is what? This one, atria is present at the junction, or I would say the end of alveolar duct, right? So many alveoli open there. Just say alveoli, it's a thin sac-like structure having simple squamous epithelium, right? Right, it is having so many blood capillaries that is giving blood to it. Obviously, that's why it is vascularized. So we have the alveolar duct as well, and so many, right? We have the alveolar duct as well, and there are so many, right? You can say a chamber is also there at the end of the alveolar duct. There is a chamber, and in that chamber, so many alveoli they will open, right? Several alveoli they will open into that atrium. So this atrium is act like a passage, or I would say collecting space of the air. Okay, a collecting space of the air. Yes or no? Yes or no? Right? So multiple alveoli, they open, they form the atria, right? The walls of atria are formed by these multiple alveoli. So this is what you should know, that alveolar atrium and the alveoli, these are the two different things. So atrium is singular, atria is plural. So this is the general thing that I told you. Now, overall, when you talk about the alveoli, right? Overall, when you talk about the alveoli, we will talk about the some small size cells here, some large size cells here. We call them as the we call them as what? We call them as the site, yes please, pneumocyte cells. Done? So let's discuss about it. So alveoli, structural and functional, right? Structural and functional unit of the lungs. Okay? 1 minute. Okay? So it is the structural and functional unit of cells, unit of lungs. It is having simple. Simple means one layered, right? We discuss it in structural organization. Simple, right? Squamous, irregular shaped, flat cells, they are simple squamous. Epithelium is lining it, right? So lined by simple squamous epithelium. So it contains many cells, like pneumocytes are there, right? You will see the cells, pneumocytes are there, macrophages, yeah, dust cells are also there. Present. Okay, so now when you talk about the pneumocyte, so you talk about pneumocyte, two types of pneumocytes are there. Do you know that? What will you see here? You will see two types of pneumocytes are there. Pneumocytes 1, pneumocytes 2. Are you aware of it? Yes, please. Are you aware of it? So we have pneumocyte. Yes, please. We have pneumocytes, right? We have the type 1, or I would say 1 and the pneumocyte 2. Is that clear? Pneumocyte 1 and pneumocyte 2. Is that clear? So this one is the pneumocyte 1. They are the smaller cells. This one is the larger cells. So this smaller cell, students, this smaller cell, how will it help? This smaller cell, my dear students, so it helps in gaseous exchange. So this is the one which actually helps in the gaseous exchange. Is that clear? This is the one that helps in the gaseous exchange. And these larger cells, what will they do? They release lecithin, right? Lecithin that acts like a surfactant. They release lecithin that acts like a surfactant. Are you getting my point? So they release lecithin that acts like what? That acts like a surfactant. This lecithin. Lecithin, I hope you know that most abundant phospholipid which is present in our body, right? It is the most abundant phospholipid. What's that? It is the most abundant phospholipids, isn't it? Isn't it? Thank you, Maria. Thank you so much. I hope you are studying well. So guys, please focus here. So what are we saying? That we have the sorry pneumocytes 1 smaller, pneumocyte 2 larger. So one will help in the gaseous exchange and the larger will release the lecithin, which is the most abundant phospholipid, right? So what will be the what will be the function of this lecithin? Yes, please. What will be the function of the lecithin? So this lecithin will reduces the surface tension of alveoli, will keep it open and will keep it open sorry, for efficient gaseous exchange. Are you getting my point? For efficient gaseous exchange. So that's how it works. This is what you should keep in your mind. Are you getting my point? Yes, please. Are you getting my point? So this is the function of the lecithin. Are you getting my point? So it reduces the surface tension of alveoli, will keep it open for the efficient gas exchange. So so larger cells, they'll release this. So this is the phospholipid. Yes, you need to know this. Okay. So this is about the alveoli. If there is any doubt here, do let me know. Okay. Do let me know. Sure. So outer side of alveoli will have, outer cell of alveoli contains fibrous tissue, yellow fibrous tissue, I would say, yellow fibrous tissue, right? And moreover, so it contains yellow fibrous tissue, number one, and in both the lungs, 300 million alveoli are present. Right, alveoli are present. Understood? So this, you should know about this detail. Okay, so that's what you should know. Is that clear? Sure. Okay, so let's start the capacities part next. So alveoli part is done. So even the NCERT part covered here. Okay, so let's move to the next topic. Let's discuss the next topic. Okay, so now we will talk about the pulmonary volumes and the capacities. So this is our next topic. This is respiratory volumes and the capacities. Is that clear? Respiratory volumes and the capacities. So how many of you, thank you Maria, how many of you love this topic and how many of you hate this topic? This topic, quick. Yes, please. How many of you consider it as a difficult one? Trust me, it's easy. We'll finish it today. I love this topic. This is easy as well. Okay, chalo, let's start. So tidal volume and respiratory capacities, or the, so how can we measure it? We can measure it with the help of spirometer. This is what you have to remember. Not all the volumes, of course, but this is what the spirometer is. Can you see that? This is what the spirometer is. Just say before surgery also, doctors, they check the lung, they check these volumes, they measure this, they check how strong the lung of that patient is, right? So this is what the spirometer is. So it is an instrument which is used to estimate the volume of air involved in breathing, which helps in clinical assessment of pulmonary function. So this instrument, right, it will help. How will it help? It will help to, it is used to estimate the volume of air which is involved in breathing, right, which helps in clinical assessment of the pulmonary function. Are you getting my point? So rate of breathing in normal healthy person, what is it? It's 12 to 16 times per minute. Are you getting my point? It's 12 to 16 times per minute. So we will discuss it. And moreover, spirometer, it cannot measure residual volume. Are you getting my point? It cannot measure residual volume. So honestly speaking, this is a topic which, if you will start reading in depth, you are going to love it. You will realize it, you know, how the running and all the things, they affect this these volumes, right? But I'll stick to the topic here. We'll share few extra interesting details, but I'll stick to the topic here. So this is what the spirometer is. So it cannot measure the residual volume. So this is what you need to know. So I will explain these tidal volumes and everything to you all, But before that, I'll go for some tricks or something. So firstly, tell me, which volumes do we need to study? Tidal volume, inspiratory reserve volume, expiratory reserve volume, and then comes the residual volume, right? What do we need to discuss? Tidal volume, inspiratory reserve volume, expiratory reserve volume, residual volume. Like 4 types of, these are the 4 volumes for respiratory or pulmonary volumes that we have to study. Okay, firstly keep it in your mind, right? So you have tidal volume, that's how you can denote inspiratory reserve volume, expiratory reserve volume, or residual volume. These are the 4 volumes that you need to study, right? Yes, please. These are the volumes that you need to study. So quickly mention it. Yes, repeat it in the chat section, write it in the chat section. Do it quickly. So this is what you have to remember. So we have tidal volume, inspiratory reserve volume, expiratory reserve volume, and the residual volume, right? And the residual volume. So that's what you have to keep in your mind first, okay? Right? So here I'll write something. So we have these 4 types of volumes. I'm repeating myself again. This is the tidal volume. Then we will discuss their values as well. Inspiratory reserve volume, expiratory reserve volume, and then comes the residual volume. Understood? This is what we have. Okay? Then here, when you talk about these pulmonary capacities, right, they are basically the combination of volumes. Different volumes together, they will make it, right? Different. So it is basically the combination of volume. Just say, let's suppose we will talk about, right? I know I have to explain you the definition. Chill, I will. Just allow me some time. Just say inspiratory capacity, expiratory capacity, inspiratory capacity, expiratory capacity, vital capacity, total lung capacity, and then we have functional residual capacity, FRC. Okay, so what do you have here? You have inspiratory capacity, expiratory capacity, vital capacity, total lung capacity, and functional residual capacity. So for these initial 4, keep tidal volume. Matlab we have to keep the tidal volume here. We'll tell you the reason, right? Now, if it is inspiratory capacity, you have to add inspiratory reserve volume. For expiratory capacity, expiratory reserve volume. For vital capacity of lungs, both ERV, IRV, plus, but when it is the total, in the total you will also include residual volume. So it is just the functional residual volume where you are just talking about the residual volume plus ERV. Otherwise, that's how it is. Firstly, you have to remember this. Are you getting my point? So, tidal volume, inspiratory reserve volume, expiratory reserve volume, residual volume. Now here we have inspiratory capacity, expiratory capacity, vital capacity, total lung capacity, or functional residual capacity. So in all these first 4, you are keeping tidal volume. So if it is inspiratory capacity, you have to add the inspiratory reserve volume, expiratory capacity, expiratory reserve volume, vital capacity mein both inspiratory as well as expiratory, or total lung capacity mein total, even you have to include the residual volume. Then comes the functional residual capacity, right? So look at it, right, and tell me if it is clear. Quick, do it quickly. Do it quickly. Yes. Done, beta? Done, students? Sure? Sure? Chalo. So let's go. Let's talk about the tidal volume. I'll explain it in detail as well. Just see, volume of air inspired or expired during a normal respiration. Like I'm talking to you, right? I'm not putting any extra efforts. Yes or no? I'm literally not putting any extra efforts. I am just, talking to you, and obviously I'm inhaling as well. So without any conscious effort, right? Without any conscious effort, air is entering in and going out. So this is what making the tidal volume, right? So tidal volume, when you are talking about the tidal volume, it is the inhalation and exhalation normally per breath, how much air you inhale or exhale without any conscious effort, or you can say quiet breath at rest. Okay, so it is the this is what tidal volume is. So you can say it is approximately 500 ml. This 500 ml is per breath. So obviously on an average, just now I told you, on an average, a healthy adult will show 12 to 16 times per breath, 12 to 16 breath per minute, right? So you can just multiply it. And you can get this. So this 6,000 to 8,000 ml of air is for per minute, right? For per minute. Are you getting my point? So 6,000 ml means 6 liter you are talking about. Now, when you talk about the tidal volume, what I just said, I told you that the value per breath will be 500 ml. Quietly, normally, where you are not putting any extra efforts, right, beta? Where you are not putting any extra efforts. So here what you will see that out of this 350 ml, it will do what? This 350 ml, it reaches the alveoli and available for gaseous exchange. This one. And this one is available for what? For gaseous exchange. And when you are talking about this 150 ml, what's this? It's the, anybody? It's the dead space volume. What do you call it? What do you call it? So it is the dead space volume, right? It is the dead space volume. Though dead space volume is of, like we talk about the anatomical dead space volume, we talk about the physiological dead space volume. So in general, we discuss that in that much detail, right? But so this is what we were talking, right? So 350 ml, it is the one which is available for the gaseous exchange, and out of this 150 ml is the dead space volume. So please tell me why this 150 ml is basically the dead space volume. Tell me quickly. Everybody, tell me quickly. Why there is a dead space volume? What is the requirement of it? Anybody, why this dead space volume exists? Yes, please. Yes? Yes, please. Why it is exists? Because more volume. No. Beta, see, dead space. Dekho, we have a passage, right? External nares, like I would say nose is there, pharynx is there, trachea is there. So these are not the sites where exchange is going on, where gaseous exchange is there. Their walls are not allowing the gaseous exchange. So this is basically the anatomical dead space volume, because here you will not see that there is any gaseous exchange, right? This oxygen and CO2 cannot diffuse through it. We know it very well. So because not all the part of this respiratory tract or the passage is for the gaseous exchange, so that's why there is the dead space volume as well. But it is not something which is useless, because it will humidify the air, it will trap the foreign particle, it will add the moisture into it, right beta? So this is not something which will get wasted, but it is necessary as well. So anatomical dead space, because here in this space, like I will talk about, nose is there, pharynx is there, trachea is there, so there is no exchange of gases. So here that air is making the dead space, anatomical dead space volume. So anatomical dead space volume is also a thing. Of physiological dead space volume is a different thing. So it is the anatomical dead space volume. Is that clear? Is that clear? So obviously this is for the normal breathing. If there will be change in that per breath, this volume will also alter. So this one you can see here, inspiratory reserve volume. So inspiration is the word here, means inhalation is the word here. So it is the additional volume of air a person can inspire. It is the additional volume a person can inspire. But how? When there is a forcible inspiration. Just why it is suggested that one should go for the pranayam or something, right? Just a minute, I'm sorry for this. So why it is suggested that one should go for yoga, these breathing exercises, how they help us and all. So basically when you are doing it, you are doing what? You are just altering these volumes at that time, now, right? Right? Yes or no? So basically this is something that you have to understand first in a simple way, that when you talk about the inspiratory reserve volume, forcibly how much you are taking in. Right? They say normally it is 500 ml, but when you are forcing yourself, it can go till 2500 to 3000 ml, and we don't use lungs properly, right? As per their efficiency. Are you getting my point? Are you getting my point? So whenever you go for the exercise or something, right? Exercise or something. So breathing, it will become very deep and rapid. The tidal volume also increases. Do you know that? Tidal volume also increases to meet that oxygen demand. More and more oxygen air will enter in our body. Is that clear? Is that clear? So yes, it is true. Thank you, beta. So yes, it is true, when we exercise, the tidal volume increasing greatly, but overall if you talk about the IRV, it will decreases, right? Right? Why is it so? I'll tell you. Just say, this is something that you can write also, that during exercise, because our body is demanding more, so tidal volume will increase. But overall, if you talk about the IRV is going to decrease during exercise. Why? Because IRV is what? Forcibly what you are inhaling. But now, whatever you are inhaling, that is because even the tidal volume demand is more, so that will go to meet out that. Are you getting my point? That will go to meet out that. Are you getting my point? So large part of this inspiratory reserve volume is used to increase the tidal volume. So during exercise, overall your tidal volume increases. Your tidal volume increases because person is already inhaling deeply. So when we go for the exercise, our breath, it becomes deep, right beta? So tidal volume will definitely increase. Okay? Okay? Done? Sure? So now I believe you know the next topic as well, that what exactly is the expiratory reserve volume. So obviously it is the additional volume of a air person can expire, right? So inspiratory mein it is inhalation, expiration mein it is exhalation. So additional volume of air a person can expire by the forcible expiration. So this is basically 1000 ml to 1100 ml. What is it? What is it? It is 1000 ml to 1100 ml. So this is something that you really need to know. So when it comes to the ERV beta, so obviously tidal volume you already know that will increase due to exercise, right? And ERV will also decrease. Okay? Okay, right? So I told you about the this explanation in this case. So ERV decreases during exercise, because large amount of it will be used to increase the tidal volume. Done. So next is residual volume. So we cannot say that our lungs, they are empty, right? Obviously you cannot say that your lungs, they are empty. The volume of air, it is in lungs. Even if after the forcible expiration, we always have the, right, we always have the air in our lungs, right? We always have the air in our lungs, right? Even after the forcible expiration, keep it in your mind, right? It's not like that your lungs are going to be empty, and this is much needed. Why? Why is it so? So first of all, its value should be clear, and even if it is more than that of your residual volume, so it is 1100 ml to 1200 ml. So it is very important. It prevent lung collapsing. So always air is there, right? Some amount of air will always be there in the lungs. So that is very important, because it will prevent our lungs from collapsing, right? Our lungs may, obviously you are talking about the alveoli here, right? Because it will keep alveoli partly inflated, right? That is their rule. They'll keep the alveoli partly inflated, right? So allow continuous gaseous exchange. It will allow continuous gaseous exchange, right? It will allow continuous gaseous exchange, and it will also make breathing smooth and efficient. Okay. And in the comment section, I want you to write why it is making the breathing smooth and efficient. Okay? Okay? Done? Sure? Any doubts? Okay? Done, beta? So that's what you have to answer. So remember this thing, that residual volume, there is no major change in the residual volume during exercise, because a part of IRV and ERV is used in increasing the tidal volume, but there is nothing, not much, or I would say no effect on the residual volume, right? So lung elasticity and all will decide this. So it is very important for our lungs to have this, because it will prevent the alveoli from collapsing. Some amount of exchange will always be there. So this is important. Now, residual volume, it can't be measured, right, by spirometer. So obviously the capacities in which residual volume is used, we cannot measure that too. So inspiratory capacity, just now I told you tidal volume plus IRV. That's how you can check. That's how you can check. So MCQs can also come on the basis of that. They can give you different values, and they can ask you that what exactly, right, what exactly will be the inspiratory capacity. Now, what is the definition of it? So total volume of air a person can inspire, because you are talking about the capacity, the ability of the lungs, right? For what? For inspiration. So total volume of air a person can inspire after a normal expiration. Normally you are exhaling, but what is the capacity of air that you can take in? That will decide your inspiratory capacity. Now, expiratory capacity, exhalation capacity, that how much air a person can expire, but you inhale normally, right? After normal inhalation. Understood? So here also we know the formula, TV plus ERV. Then after that, vital capacity, we'll discuss it first. So maximum volume of air a person can breathe in after a forced expiration, right? Like now I have to check the capacity of my lungs, right? But forcibly I have to expire, so that later on I can check that how much, right, can enter, right? In both the cases we can check this, right? For both the cases we can check this. What? We can check this particular capacity. So when it is the vital capacity, we also check maximum volume we can breathe in after a forced expiration, or, right, maximum volume a person can breathe in after a forced expiration, or maximum volume a person can breathe out after a forced inspiration. So that's how we can check it. Okay, so you know that. So that is what it is. Understood? And then comes the total. So total lung capacity means volume of air which is accommodated in the lungs, right? But how? At the end of forced inspiration. So it is tidal volume plus IRV plus ERV plus residual volume, and we know that these three things are equals to vital capacity. So it is vital capacity plus residual volume. So in both the ways you can write it, in both the ways. Okay, so functional residual capacity is the volume of air that is in the lungs even after normal expiration, after a normal expiration. So you know what it is. So if we cannot check residual volume, we cannot check these two as well. Got that? Right? So some teachers, they explain it with this chart also, right? That this one is the tidal volume per breath. Vital capacity will include your tidal volume plus this inspiratory reserve volume and expiratory one. So this is what I have added, but in a very simple way you guys can remember it. Okay, so this one though we have to discuss yet. Chalo, answer this question again, a very simple one. Guys, quick, answer that question. Done, beta? So answer this question quick. Yes, please. So let's talk about the next part. So this one is expiratory capacity, this one is total, and this one is functional residual capacity. Chaliye this one, solve it quick, solve it quick. Guys, quick. Beta, this is for those who are in 11th and they think they have wasted it. Even anybody can watch it because we are delivering everything, right? You are getting all the content. So do not worry about which series it is, right? Just focus on the content and revise the topic. So this one, quick, answer it, then we'll start the mechanism of breathing. Okay, voice low, I think it's fine. Just check it out. Yes, please, what's your doubt? So this one, then inspiratory reserve volume. Yes, please. So expiratory one, residual one, and then you can just check the vital capacity. Yes or no? Okay. Done? Chaliye. So next, this one, answer it, quick. Answer it, everyone, quick. Quick, quick, quick. Answer it. Then we'll start the next part. It's a very easy question. Then after it, you will go for mechanism of breathing. That's what we have to understand. This is what are they saying? They are saying that inspiratory reserve volume plus tidal volume plus expiratory reserve volume. So you are talking about the vital capacity here, isn't it? You are talking about the vital capacity here, isn't it? So this is equivalent to, yes please. So if I see this part, yes, I can say inspiratory capacity plus expiratory reserve volume. True that. True that, है ना? But there is no functional residual capacity. We cannot consider this. We cannot consider this. Cannot consider it. Yes, right? So you know that because IRV and TV, this is equals to inspiratory capacity. So that's how you can check it. Got that? Sure? Okay. So the next part that we have to understand is the mechanism of breathing. But before that, just have a look. Here the diagrams, I told you right lung is having 3 lobes, horizontal fissure is there, oblique fissure is there, but in this case you just have oblique fissure. So that's why left lung will show you only 2 lobes and this is the cardiac notch, right? This is the cardiac notch and these are the alveoli, right? You can see they have the abundant blood supply. And now let's start the mechanism of breathing, okay? Let's start what? Let's start the mechanism of breathing. So here in breathing, we'll talk about the inspiration and we will also talk about the expiration. Isn't it? This is what mechanism of breathing is. So inspiration is also known as inhalation. Expiration is also known as exhalation. है ना? Inspiration is also known as inhalation. It is also known as exhalation. Are you getting my point? है ना? So that's what we have to discuss. So before starting the mechanism of breathing, we have to discuss few basics. Then we will go ahead. Okay? Okay, so but one thing that you have to understand, that this is the airtight chamber in which our lungs are placed. Okay, in which our lungs are placed. So see, first of all, you know, we have to understand a concept of diffusion. So what exactly is the diffusion? What exactly is the diffusion? Tell me. Quick. What exactly is the diffusion? Basically, when you say diffusion of gases, so it is gases moving from their higher concentration, or I would say higher partial pressure, to lower one. That's how we revise it, yes or no? Right? In the case of gases, like basically diffusion, what exactly it is? It is the gases basically, you know, that they are moving from their higher concentration to the lower, or higher partial pressure to the lower pressure. This is what the diffusion of gases is. Now, if I have to inhale, right, if I want the air to come inside, right, now I'm using the word air here. I'm not saying oxygen only. We need the oxygen, but we know that air is not only oxygen. Air is actually the mixture of gases. Air is having oxygen, nitrogen, it is also having the CO2, etc., etc. So air, which is a mixture of gases, so how that air will enter in our lungs? If the atmospheric air, right, if externally, if in atmosphere, air, if its pressure is more than that of the lungs, in lungs if it is less. But before knowing this thing, understand one thing. Air, as I said, it is a mixture of gases. It is a mixture of gases. So obviously each gas is having a pressure here. Each gas is having a pressure here. Isn't it? Like if I talk about the total pressure of the air, it is equals to the pressure of all the gases. So each gas, each component is having its own pressure. So when you talk about the pressure of that each component, you are using the word partial pressure. Like for an example, you're talking about the oxygen. So here you will not say pressure of oxygen in air. You will talk about the partial pressure of oxygen, right? Partial pressure of oxygen in air, because air is a mixture of gases. Air is made up of many gases, and each gas will contribute. Each gas will contribute in its pressure. And if I have to talk about the pressure of each gas, so I will use the word partial pressure, isn't it? What is the term? What is the word that I will use? I will call it as the partial pressure. Are you getting my point? What will I call it? I will call it as the partial pressure, my dear students. Okay. Okay, so each gas here will exert a pressure, and that word, for that we will say it is the partial pressure. So if we have to check the pressure of air, we have to add the pressure of all the gases present in it. Is that clear? So now when we inhale, so we inhale the air, and what do you need? You need the oxygen, because that oxygen is going to oxidize your food, isn't it? That oxygen is going to do the respiration. Are you getting my point? So we say partial pressure of oxygen. So if we want to inhale, if we want the oxygen to enter in our body, then what we need, that in the air, if partial pressure, and this is what diffusion is, if partial pressure of oxygen, if it will be more in the air, right, and if in the lungs, if the partial pressure of oxygen is less, then only oxygen will come from outside to inside. Now, yeah, air will come from outside to inside. So if the pressure of air in the atmosphere is more, and if in the lungs it is less, then only it will move. So lungs, when they inhale, they have negative within intrapulmonary pressure. Right? They have negative intrapulmonary pressure. That's what you have to remember. है ना? But are we going to start it from this part only? No. We have to know one thing that is known as Boyle's law. So this Boyle's law is applicable here. right? Then I will talk about the muscles. Firstly, understand this. As per this law, volume and pressure, as per this law, volume and pressure, they are inversely proportional to each other. They are what? They are inversely proportional to each other. What? This volume and pressure. They are what? They are inversely proportional to each other. They are not directly proportional. So it means if the volume increases, pressure decreases. If pressure increases, volume decreases. Are you getting my point? This is what it means. And this is applicable here. Like if I want the air to come in the lungs, I have to decrease its, the pressure should be less. Outside pressure should be more. So how this pressure will be less? If volume is more. This is what Boyle's law. If we want the air to enter in the lungs, right? So if we want less pressure, we want less pressure. So for less pressure, we need to increase the volume of the lungs. We need to increase the volume of the lungs. Now, you know it very well, right? You know it very well that lungs, where are they present? They are present in anatomical airtight chamber, right? Where are they present? They are present in the anatomical airtight chamber. I believe you all know that. I believe you all know that. You know that lungs, they don't have their muscle. They cannot regulate their volume by itself, right? Lungs are not reducing or increasing their volume. We know that lungs are placed in the thoracic cavity in such a way that if there is any change in the thoracic volume, that will reflect as a change in the pulmonary volume, right? That will reflect as a change in the pulmonary volume. If I am, because thoracic cavity, when I'm talking about the thoracic cavity, this is something which I have to see, right? See this. This is what I have. I know that in the thoracic cavity here, we have dome-shaped diaphragm, which is again, right? Peripheral part is basically the skeletal muscle there, right? We know that here the sternum, ribs, and here particularly thoracic vertebrae, it is present. So when I look at this part, this anatomical airtight chamber, I know that here I have this dome-shaped diaphragm, which is dividing this thoracic and abdominal cavity. Moreover, if you pay attention, you know that in between these ribs, can you see this part here? In between these ribs, can you see this part here? Right, in between these ribs, you also talk about the intercostal muscles. What do you discuss here? You talk about the intercostal muscles, right? Which muscles are we talking? Intercostal muscles, intercostal muscles. So during this complete breathing mechanism, which will involve inspiration as well as expiration, we'll be talking about the intercostal muscles, this diaphragm muscles even, because forcible, sometimes this forcible expiration is also there. Abdominal muscles also play the role. So we will talk about them. So firstly, let's start from this part, right? So very first thing that you need to understand here is, see, I told you, as I said, these are the ribs, and in between ribs, we have the muscles. And look at this part, right? Just say this is our rib cage. This is our rib cage here. We are cutting it. We are dissecting it, right? So when you look at the diagram in the NCERT, when you look at the diagram in the NCERT, just see this. So we see ribs and the sternum, right? This is what they are showing. This is showing the rib cage. So it is just like you have dissected it and then you are observing this part, right? Let me show you this. See this. This is the cut end of the ribs, because you are taking a section, you are cutting the ribs as well. So this is showing the cut end of the ribs. And look at the diaphragm, right? Look at the diaphragm part here. So I hope this part is clear. So now, yes, so this is what we have. So when you are talking about this inspiration and inhalation, remember it is an active process. It is an active process where you use the energy. And this one is going to be what? This is a passive process, right? If it is not forcible, then it is what? Then it is the passive process. Is that clear? Then it is what, beta? Then it is the passive process. So this is the first thing that you have to keep in your mind. So when you talk about the inspiration, it is for 2 seconds, right? It is for 2 seconds, and this one will be for 3 seconds. Understood? This one will be for 3 seconds. So inspiration is an active process, but when you talk about the, right, when you talk about the expiration, then it is going to be what? It is the passive process. Got that? Right? Right, so muscles are going to play the role. So that's what we have to discuss. So basically, let's talk about the inspiration. Right, basically we are talking about the inspiration, but before that, just see, these are the ribs. So I told you they have intercostal muscles, means they are present in between the ribs. Now when you look at it, just let's suppose these are the ribs. So the muscles which are present on the external side, can I call them as external intercostal muscles? Definitely. And the muscles which will be on the internal side, I will call them as internal intercostal muscles, right? So in the inspiration, right, during inspiration, what exactly you have to understand? We will talk about the diaphragm muscles, and there we will talk about the external intercostal muscles. It is EICM, right? So these two are considered as the inspiratory muscles. Again a PYQ. So these two are considered as what? They are considered as the inspiratory muscles. So this is literally a PYQ. Why do you call them as inspiratory muscles? Because they will undergo contraction during inspiration. These are the muscles that undergo contraction during inspiration. That's why you call them as inspiratory muscles, because they will undergo contraction. So which muscles are they? Diaphragm muscles and external intercostal muscles. Now let's understand this part. You know that diaphragm is dome shaped, right? Diaphragm is what? Diaphragm is dome shaped, right? Now what will happen? So during inhalation, peripheral muscles of this diaphragm, they contract. What will happen? Diaphragm muscles, they contract, due to which diaphragm becomes dome shaped, sorry, flat. This is important. Again, diaphragm is dome shaped when there is, like in a normal state, when you are not inhaling, right? During inhalation, diaphragm muscles contract, due to which diaphragm becomes flat. Now understand this part. Yes, so I hope it is visible to all of you. So now understand this part. Just say, imagine if this is your thoracic cavity, right? If this is the thoracic cavity, this is the thoracic cavity. Yes or no? So when diaphragm is dome shaped, now we know that it will become now when it will become flat, then don't you think so diaphragm muscle contract. So we know that here diaphragm is what? Diaphragm is flat, and due to which what will happen, students? Volume towards anterior posterior axis increases. Yes or no? That is what you have to remember. Done? Okay. So diaphragm muscles, external intercostal muscles, they are the inspiratory muscles because they will contract during inspiration. So diaphragm normally is dome shaped, but due to inhalation, its muscles will contract. Diaphragm will become flat shaped. So when diaphragm is flat, what will happen? Its volume will increase towards the anterior posterior axis. Are you getting my point? Its volume will increase towards the anterior posterior axis. That's what you have to keep in your mind. Number one. Secondly, now when you are talking about the muscles here, right? You know that now when you are talking about the external intercostal muscles, now you have to understand when they contract what will happen. Understand this thing. Just say this is what you have to remember. I always say for breathing, these are the two dance steps which we have to keep in our mind. First, you have to keep your hand like this, then this, 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 this. Then it is like this, this, this. So diaphragm, dome shaped, become flat. Volume towards anterior posterior axis increasing. Your rib cage, it is inward, downward. When the external intercostal muscle contract, it will go outward, upward. So external intercostal muscles, when they contract, the rib cage move upward and outward. Outward. Just normally you can see that external costal muscles normally relaxed. So when they are relaxed, guys, what will happen? The rib cage is downward, inward. Done? And so when these muscles they contract, your rib cage will move upward and outward. So now you understand this thing. Due to which what will happen? Volume towards dorsoventral axis increases. So what will happen here? Volume towards, right? Yes, please. Volume towards dorsoventrally and laterally even increases, right? Right? So overall, during inspiration, if your rib cage was like this, so during this inspiration, that's going to happen. Basically, volume towards anterior posterior axis and this one will be due to diaphragm muscles and dorsoventral due to external intercostal muscles. Understood? So this is happening during inspiration, which is an active process. I hope it's done. I hope it's done. So any doubts here? Quick. Any doubts here? Any doubts here? Quick, if there is any doubt, do let me know. Okay, so obviously when there is the expiration, that is exhalation, right? So this is passive for 3 seconds, and here what's going to happen? Your external intercostal muscle relax. If it will relax, we know that the rib cage comes downward and inward. And your diaphragm muscles relax. Then diaphragm becomes dome shaped. You know it very well, right? So here we know that volume decreases and intrapulmonary pressure increases, isn't it? Then the exhalation occurs. And during inspiration, you know that there will be negative. Intra means within, intrapulmonary pressure. And here you can see Boyle's law is completely applicable because volume increases, pressure decreases. So you can say negative intrapulmonary pressure, or you can say that intrapulmonary pressure is lesser than the outside. So here volume will decrease, intrapulmonary pressure will increase. So this is going to happen here. So this is for the expiration. So if there is the forcible expiration, we know it very well, right? Right. So during forcible expiration, when forcefully we exhale like this, then internal intercostal muscles and abdominal muscles they contract. They contract. Is that clear? So they contract, and we know that that contraction will result in decrease in volume, increase in pressure. Understood? And forcible expiration is active. Is active. Understood? Is active. Write down. Quick. Done, students? Sure? Write down quickly. Okay, so let's done. Okay, so this is the part. Now, there are a few other points that I would like to discuss here. Then we'll talk about the thoracic and the abdominal respiration. So here you can see. So inhalation is active, muscles contract. Exhalation is passive, muscles relax. But if there is forceful exhalation, the muscles, internal intercostal muscle and the abdominal muscles, they contract. So abdominal muscles, they increases that efficiency as well. So here you can see during inhalation, ribs move up and out, right? So the volume towards the dorsoventral axis will increase and the diaphragm curve down, it becomes flat. So the volume towards the anterior posterior axis increases, right? So other than that, yes, so here you can see, right, the diagram again. So diaphragm is contracted, so this arrow is showing it is going downward. So here you can see air is entering the lungs, ribs and sternum is raised, volume of thoracic increased. This is what rib cage is, and here you can see diaphragm is relaxed, arched upwards, air is expelled from the legs, ribs and lungs, ribs and sternum return to the original position, volume of thorax decrease. So diagram should be clear. Are you getting my point? So the diagram should be very clear to all of you. Okay, so this is again a comparison table that I have added. So see, inspiration occurs when intrapulmonary pressure becomes less than atmospheric pressure, lungs have negative pressure. Expiration occurs when intrapulmonary pressure becomes higher than atmospheric pressure. Inhalation active, exhalation when normally it is passive. It is initiated by contraction of diaphragm, diaphragm flat, thoracic cavity increases anterior posteriorly, diaphragm here relax, returns to dome shaped. So question can come on the basis of the shape of diaphragm during inhalation and the exhalation. Then external intercostal muscles contract, ribs and sternum move upward, forward and outward, increasing thoracic volume dorsoventrally and laterally. External intercostal muscles relax, ribs and sternum return to the normal position. Here there is increase in thoracic volume, and we know that if there is increase in thoracic volume, there will be increase in pulmonary volume and there will be decrease in intrapulmonary pressure, air enters the lungs. Here there is decrease in thoracic volume, increase in intrapulmonary pressure, air is expelled from the lungs. So in forceful inspiration, additional muscles may assist. In forceful expiration, internal intercostal muscle and abdominal muscles contract. So that is what I have explained with the help of this table. And when you read the NCERT part, you can see this thing very clearly here. We have the ability to increase the strength of the inspiration and expiration with the help of additional muscles in the abdomen. So on an average, a healthy human breathes 12 to 16 times per minute. So volume of air involved in breathing movement may be estimated by using spirometer, which we have already discussed and understood. So this table is also, I hope this table is also clear to you. So next part is the exchange of gases, which we have to discuss. So here in the notes part, I have added these short points also. Ki active hai, pharynx muscles relax, external intercostal muscles relax. So these are the points that I have discussed. So the next topic is the exchange of gases. Understood? Then we will talk about the transport of gases. Okay. So any doubts so far? Any doubts so far? Any doubts so far? Quick. Sure. So now we will talk about, so this part is done, right? Okay, so now the next thing that we all have to discuss is the thoracic, yes please, thoracic and the abdominal breathing. These are the two types. So do you know about them? Yes? Do you know about them? Thoracic and the abdominal breathing? So normally it is the abdominal breathing, right? So when you talk about the thoracic or the costal breathing, so external intercostal muscles, they contract, the same thing is there, right? So that this thoracic breathing is more prominent in females, right? So abdominal breathing is basically the diaphragm breathing, right? So you know that what will happen to the diaphragm. So more prominent in the males, seen in infants, more efficient and energy saving. So this is about these two types of breathing, right? And in normal quiet breathing, right? In normal quiet breathing, you will see both types occur together, but one predominates the other. It depends upon the age, sex, and the condition of the body. It's like that. Okay, it's like that. Now the next topic is the exchange of gases, and we know that this thing will also occur due to the diffusion, and we know that here also there will be the difference of the partial pressure. So that is what we have to discuss next. Now students, when you look at this part, ki alveoli, they are the primary site for the exchange of gases, already told you. So exchange will occur here between the blood and the tissue. So this exchange that is taking place here, right, between the blood and the tissue, right, between the blood and the tissue. So oxygen and CO2 are exchanged here, right? We know that by simple diffusion. So it depends on what? It depends on the pressure or the concentration gradient. Okay, solubility. So chalo, one by one we will discuss it. So tell me, up to this part all clear? Up to this part all clear? So we know that it is due to the diffusion will occur. There are factors which will decide the diffusion thing, right? That what will be the rate of diffusion. So overall, if in general, firstly discuss, diffusion will occur. So how is it possible? Obviously, when it comes to the diffusion, solubility of the gas will be very important factor, that how soluble the gas is. Like if I say CO2 is more soluble, right? CO2 is more soluble than oxygen. Do you know that? This is very important. Secondly, this thickness of the membrane. If the membrane will be very thick, the diffusion will be affected, right? Right. So even the partial pressure thing, this solubility thing, and this thickness and thinness, we have to consider when we talk about what? When we talk about the diffusion, right? When we talk about what? When we talk about that diffusion. Is that clear? Is that clear? So I will not dive deep into this topic. We'll stick to the NCERT part here, right? So when it comes to the, I'm writing it here that for diffusion, what are the factors? The rate of diffusion will be decided by obviously pressure / concentration gradient of that gas. Secondly, solubility. As I said, CO2 is more soluble than oxygen. Then it should be the thickness, or I would say thinness of the membrane, right? So when you talk about the exchange here, the respiratory membrane here, it should be very, very, very thin, less than 0.1 mm, right? So let's discuss it. Is that clear? So see this. Firstly, here, we know that this is the external respiration. I hope you are aware of it. What's that? It is the external respiration, right? When there is the exchange in between the organism and the environment, this is the external respiration. Here we know that this is the alveoli, primary site of exchange of gases. This is the blood capillary. This is what? This is the blood capillary. So this exchange is actually the external respiration. Okay, this is what? This is the external respiration. So let's discuss about it. Right, let's discuss about it. So up to this part all clear? Up to this part all clear? Yes? Done, students? So these are the factors which are given. Baki we know that it depends on the size as well. We know that it depends on the medium as well. Diffusion is more in gases, right? We know it very well. And moreover, temperature. So if there is increase in temperature, even the diffusion will also increases. Is that clear? Is that clear? Right? These are the basics. And I believe you all know that. And I believe you all know that. So rate of diffusion, it depends upon the diffusion coefficient, beta, right, which will depend on the nature of gas, the medium, even the temperature, even the molecular size. Area is also important, right? More surface area, faster will be the diffusion. And other than that, we know that concentration gradient and thinness will be there. But that formula, that calculations, not required for the NEET examination. Is that clear? So now, when you talk about this part, see, we know it very well, right? Even in the structural organization, we have studied this. This is simple, mane, single layer. Like when we talk about the types of epithelial tissue, right? When we talk about what? When we talk about the types of epithelial tissue, we discuss the simple epithelium, we discuss the compound epithelium. So whenever we see, right, what are we talking about? We are talking about the epithelial tissue. Whenever we say it is a simple epithelial tissue, it can be compound, further types are also there. So simple mane single layered. Simple mane what? Simple mane single layered. And here that is uniserrated. Okay. And here you have other things also, like what type of cell is there. There can be squamous, right, where irregularly flat shaped cells are there, also known as pavement epithelium, tessellated epithelium. The cells can be cuboidal. We study that in class 9th also. We have columnar epithelium as well. But whenever you talk about the sites where the diffusion, right, will occur, you will find that it is the simple squamous epithelium. So now when you talk about the alveoli, right, so alveoli is lined with simple, mane single layer, squamous, mane flat, irregularly shaped cell. So simple squamous epithelium. It is lined with what? It is lined with simple squamous epithelium, right, students? And we know that epithelial tissue, it lies on what? It lies on what? We know it very well that epithelial tissue, it lies on the basement membrane. It lies on what? Basement membrane. Is that clear? Then after that, see, this is the blood capillary. Right? So blood capillary, very thin. So it is having, we call it as what? Endothelium. Right? Endothelium of blood capillary. So it is also the simple squamous epithelial tissue. Are you getting my point? Right? It is also the simple squamous epithelial tissue. Is that clear? And basement membrane in two. Basement membrane present in two. In these two. So this is together making these layers together making the respiratory membrane through which the exchange will occur. So if it is very thick, the diffusion will be slow. If it is thin, it will be fast. Is that clear? So have a look, right? Have a look. Just now told you that simple diffusion, through simple diffusion, this exchange will occur, which is based on number one, right? Pressure and the concentration gradient. Number two, solubility of the gases, as well as number three, thickness of the membrane. Okay, so pressure contributed by an individual gas, because we know that air is the mixture of gases. Air is what? Air is the mixture of gases. Air is what? Air is the mixture of gases, and here each gas will contribute to that pressure. So we will use the word partial pressure. So PO2 means partial pressure for oxygen, partial pressure of CO2, right, students? So here you can see we have this table, very important one, the one from where every time they ask the question, right? So look at this, respiratory gases, atmospheric air. So this pressure is in mmHg. So 159 mmHg. So what's that? It is 159 mmHg, right? But in alveoli, it is 104 mmHg. In the deoxygenated blood, it doesn't mean it doesn't have oxygen at all. No, it means it is having less partial pressure of oxygen in comparison to the oxygenated blood. But you cannot say that if it is the deoxygenated blood, it doesn't have oxygen at all. Are you getting my point? Are you getting my point? And then comes the tissue. So tissue is having what? Tissue is having 40 mm of Hg. So yes, we need to remember this. So CO2, you know that atmospheric air 0.3 mm Hg, alveoli में 40 mm Hg, where the oxygen's partial pressure is 104. Here in the deoxygenated blood, look at this. Deoxygenated blood will have 45 mm of Hg of CO2 partial pressure, and here 40. So if there is increase, so this is what we have then. Blood, oxygenated blood 95 and oxygenated blood 40. 40 is the partial pressure. So if there is an increase in the partial pressure of CO2 in this arterial blood, right, then it indicates that inspiration is required. And look at the tissue part. Now, before going forward, I would like to ask you this thing. Please tell me, everybody, please tell me why this difference is there. In the atmospheric air, the partial pressure is 159 mmHg, but in alveoli it is 104. Please tell me why. Guys, quick. I really want to know this. Please tell me why. Quick. Yes, please, I'm waiting for your answer, guys, quick. Why is it so? Here, in atmospheric air, it is 159 mmHg. But in alveoli, the partial pressure is 104 mmHg. Why is it so? Why there is that difference? Why? See, these are the reasons here. You guys can see. Atmospheric air and alveolar air, we know that they have different partial pressure. We have inhaled that air at that partial pressure, but in alveoli, its partial pressure is less. Why is it so? Because air in alveoli is not fresh atmospheric air. By the time it reaches the alveoli, there are some changes. It will mix with the residual air there. You know that residual air is present there, right? You know that residual air is there in our lungs. So yes, it will mix with the residual air. In every breath, the inhaled air will mix with that air. So the alveolar air is always a mixture of old and new air. That is going to affect the partial pressure. Then gases exchange in alveoli is also taking place. Continuously oxygen is diffusing out from the alveoli. CO2 is continuously coming in. Right? So this is going on. That will definitely going to lower the partial pressure of oxygen because CO2 concentration is also increasing in the alveolar air. Then humidification. Air will be fully saturated with water vapors in the respiratory tract. So water vapor, they will exert its own pressure, reducing the partial pressure available for oxygen and other gases. So that is why you see this difference here, that in atmospheric air it is 159 mmHg, here it is 104 mmHg, and these are the reasons which are listed here. Can you see that? Yes, please. Can you see that? So, as I said, mixing with the residual air, gas exchange plus humidification is also there, right? Air will be fully saturated with water vapor in the respiratory tract that will have their own pressure. That will decrease the partial pressure of the oxygen here. Is there any doubt? Right? Nobody will explain you these things in that much depth, beta, and you have to read, you have to revise, you have to study your NCERT in this way only, right? Where you should know what is written here, what is written here, the differences and everything. You people should be clear, right? Okay. So now let's see this part. See this diagram also, they have mentioned it. So partial pressure of oxygen 104 mmHg, CO2 40 mmHg. So this is given here. So now we'll discuss the next part, right? I'll come to this part later, right? I'll discuss it in this in depth also. So firstly, let's talk about it. Chali. So now, you know that, right? After this table, what was our last line before this table? So similarly, a gradient in pressure for CO2 is in the opposite direction, that is tissue to blood and blood to alveoli. So solubility of CO2, we have already talked about it, 20 to 25 times higher than that of the oxygen. Okay, so this means, what does it mean? It simply means that CO2 that can diffuse through the diffusion membrane per unit difference in partial pressure is high as that of the O2. I told you solubility is something important. So solubility of CO2 is more, so more CO2 will diffuse through this diffusion membrane. Is that clear? Is that clear? right? So diffusion membrane में या respiratory membrane में 3 major layers are there. Again discuss thin squamous epithelium of alveoli, endothelium of alveolar capillary, it is again the simple squamous epithelium only, and the basement substance or the basement membrane which is supporting this, it is also present, right? It is also present. So this is something very important, guys. So as I said, so its thickness should be less than 1 millimeter, right? I think initially I mentioned it 0.1. No, it should be less than 1 millimeter. So correction here. So it should be less than 1 millimeter. So therefore, all the factors in our body are favorable for the diffusion of oxygen from alveoli to tissue and CO2 from tissue to alveoli. So that's what you have to keep in your mind. Am I clear? Am I clear, everyone? So see the diagram here. Squamous epithelium of alveolar wall, one-celled thick. Basement substance present in these two, which is supporting this and this layer. Endothelium of blood capillaries, and these are the RBCs. So multiple times they have asked this in the exam as well. So you should know the details, the diagram, the description, read everything carefully while reading the NCERT. Understood? While reading the NCERT. So the next topic is the transport of gases, but before that, see, you know that in the case of heart, we have a trick. Like, I'll finish this part. Now, few terms here. Lord. Lord means, we will make it lard as well, that we will discuss later. Left side of the heart is having the oxygenated blood, and right side of the heart is having the deoxygenated blood. This is one thing that we all know. When we talk about this, we know that heart is a pumping organ. We know that arteries, they carry the oxygenated blood, the blood in which the partial pressure of oxygen is more. But yes, we do have an exception. We do have an exception. What's that exception? That exception is pulmonary artery. So pulmonary word is associated with lungs. So pulmonary artery, it carry deoxygenated blood. It carry deoxygenated blood. This is number one thing. Now, veins, what do they carry? They carry deoxygenated blood, right? Here, what's the exception? The exception is simple, pulmonary vein. So, pulmonary vein, it carry oxygenated blood, right? It carries oxygenated blood. Am I clear? Now, you know that left side of the heart is going to pump oxygenated blood, but how? Basically, what is happening? This is the, we know that in a heart, right auricle, right ventricle is there, left auricle, left ventricle is there. We are well aware of it, isn't it? Now we know that right side will carry the deoxygenated blood. So all the vena cavas, you know, coronary sinus, superior, inferior vena cava, they all will bring the deoxygenated blood to the right auricle. It will pass to the right ventricle. Then from the right ventricle, it is the pulmonary artery, because I told you already that it is an exception. So it will carry it to the lungs. I hope you all understand that, right? We know that our heart is 4-chambered. We know that right side, right auricle will receive from all the systematic veins, or you can say inferior, superior vena cava, coronary sinuses, they will give the coronary veins, they will give the deoxygenated blood here. It will go to the right ventricle. So right ventricle with the help of, as I said here, pulmonary artery, because this is an exception. Clearly, I have mentioned it here. So this will carry the deoxygenated blood to the lungs. So what that lungs will do, they do the oxygenation, not oxidation. Oxidation is different. Oxidation, we say that when we say na, that oxygen is used to, you know, oxidize the glucose, right? But here you are saying oxygenation, meaning addition of oxygen. Lungs will do the oxygenation, and then with the help of pulmonary vein, because pulmonary word is associated with your lungs. So with the help of pulmonary veins, the blood will pass to the left auricle, then to the left ventricle. Then we know that it is the dorsal aorta or the systemic aorta that will carry it to the different body parts. So this is the diagram that I have explained you. I have explained you here. This is the external respiration. This one is the internal respiration. So I hope this diagram is also clear. So when you will study the structure of heart, then you'll have more clarity of it. Am I clear? Am I clear? Then after this, we have to discuss the transport of oxygen and the transport of CO2. So up to this part, all clear? Then the next part will be the transport. Done. Diagram clear? So we are not going to leave even a single line from the NCERT. Don't worry about that. Okay, main topic is the... Okay, so now let's speed up because main topic is this transport. Then we will talk about the regulation. Disorders are easy. So let's finish it, say. Or do you want part 2? Huh? Do you want part 2 or is that okay? Huh? Quick. Part 2 or is it okay? Yes, everyone? Quick. Ready to finish it today or you want part 2? Huh? Whatever you say. Let's continue. Okay, so let's finish it. Let's start it. So let's talk about the transport of gases. Ready? Chalo. Let's go. So transport of gases we have to discuss. We know that it is the blood which is the medium for the transport of gases. So let's discuss it individually. First, let's talk about the transport of oxygen. Okay? Let's talk about the transport of oxygen. But firstly, let's have 5 minutes ka break. Then I will continue. Okay? Let's have the 5 minutes ka break. Okay? Just 5 minutes and then we will continue. Allow me one. Ready, guys? Hello, let's start again. Uh-oh, where is my pen? Got that. Chalo, let's start again. So we are going to start with what? We are going to start with the transport of oxygen. So students, when we talk about the transport of oxygen, I hope I'm audible, right? So when we talk about the transport of oxygen, so 3% of oxygen is transported in dissolved form, transported as dissolved form in plasma. In plasma. Am I clear? As the dissolved form in plasma. What? Your 3%, right? Your 3% will be transported like that. But when you talk about the 90% of the oxygen, 90% of the oxygen will be transported in the form of oxyhemoglobin. In the form of what? Oxyhemoglobin. So this is what we have to discuss next. So when you talk about the hemoglobin, what exactly is the hemoglobin? We know that hemoglobin is present in RBC, right? It is the respiratory pigment. It is present in RBC, it is the respiratory pigment, and it's a quaternary protein, right? Right, what is it? It is also a quaternary protein. Yes or no? Yes or no? What? The hemoglobin, right? So now see, we are mainly discussing the second last topic, then we will talk about the regulation of respiration, right? So rest is easy, so we have to discuss the quaternary, so we have to discuss this part, and this is very easy, trust me on that. So it's the quaternary protein. So basically what does it means? So it means that here we will see that 4 polypeptide chains are present. How many chains are present? 4 polypeptide chains are present. Basically in adult hemoglobin, you will see that there are 2 alpha chains and 2 beta chains. Let me tell you, beta, fetal hemoglobin and the adult hemoglobin both are 2 different things. Adult hemoglobin and the fetal hemoglobin, they are different, their structure is different. Fetal hemoglobin is having more affinity for oxygen. Even if there is low partial pressure, fetal hemoglobin is having more affinity for the oxygen in comparison to the adult one. So 2 alpha, 2 beta chains are there, right? So this is what your quaternary protein is. So in the case of hemoglobin, it is the globin. It is the globin which is the protein part. So heme is the pigment here, right? Heme is what? Heme is the pigment here. So just look at this. Right, just look at this. So this is the hemoglobin, 4 polypeptide chain, and here you will see this is the heme part having the iron, and it is Fe2 positive. What's that? It is Fe2 positive, right? So iron is present here. That's why we say that iron is very important. You know, if there is a deficiency of iron, anemia can occur. So that's what you have to understand. So it is the Fe2 positive, means your iron is in +2 oxidation state. So we call it as ferrous. So basically there are 4 polypeptide chain, right? Globin is the protein part, heme is the pigment here, and when you talk about it, right, this heme, what will you see here? You'll see the iron, right, the central metal iron. So it is in it is the ferrous basically, right? So it is in this +2 oxidation state. That's what you have to remember. Now one more thing, when you talk about this is the first thing. So this part is clear. This part is clear. Now what's the next part? So basically oxygen and hemoglobin, they combine. Oxygen and hemoglobin, they combine. So what do we call it? They combine. What do we call it? We call it as oxygenation. Now we have to understand how do they combine, what happened, what are the condition. But what is the word for it? It is oxygenation. What is the meaning of oxygenation? Oxygenation mane addition of oxygen. What is the meaning of oxygenation, guys? It means addition of oxygen. Oxygen will be added here. It is not oxidation, right? The oxygen is not going to oxidize the hemoglobin. It is the oxygenation. This is the addition of oxygen, right? Now, we know one thing, right? We know one thing, that hemoglobin is a respiratory pigment. It basically transport oxygen. It basically transport oxygen. Now, listen to me very carefully. If it has to transport the oxygen, do you think it can have a permanent relationship with it? Hemoglobin and oxygen can have a permanent relationship. You take it like you consider it like a delivery person, right? Whenever we receive any of our order, any of our parcel, so basically what is it? Like from a delivery center, the delivery person will pick that up and will give it to us. So delivery person is somebody who's transporting it, who's giving it to us, who's right? So it's not like that. Like let's suppose you have ordered let's suppose you have ordered a mobile phone. So delivery person is going to pick that up and will give it to you, right? Delivery person is not going to own it. So that relationship is temporary, right? That relationship is temporary, and that should be the thing. So same is the case here, right? Same is the case here. So combination of oxygen with hemoglobin is oxygenation, right? So during oxygenation, the bond formation between oxygen and hemoglobin is temporary and reversible. So it's not something permanent, it's temporary and reversible. So we know that, see, one hemoglobin, right? It is having heme groups, 4 heme groups, right? And one heme group can join with one. Like let's see, this is the diagram here. This is Fe2 positive. It can join with one oxygen. So this can also join with one oxygen. This can also join with one oxygen. This can also join with one oxygen, right? Right. So when hemoglobin joins with O2, right? Right, when joins with O2, this is the oxygenation. What is it? It is the oxygenation. Is that clear? Is that clear? Right? So when hemoglobin joins with first oxygen, it increases its affinity for next oxygen molecule. Yes, right? It is going to do that. Keep it in your mind, okay? Keep it in your mind. So this will continue till all 4 hemes are saturated. This will continue till all 4 heme groups are saturated. Told you already about it. Yes or no? Yes or no? So one hemoglobin can join with 4 oxygen molecule, right, students? So it is Hb4 4O2. So it is Hb4O8. That's how you can write it. Is that clear? So this is your oxyhemoglobin, which forms temporarily and in a reversible manner. So on what factors it depends? So it depends on, so this is important, right? It depends on, understand this thing, because that will help you to understand the oxygen hemoglobin dissociation curve. Is that clear? Is that clear? So it depends on partial pressure of oxygen. See, what favors it? That's what you have to remember. What favors it and what is not going to favor it? Let's talk about that, right? Let's talk about that. So, okay, so when there is high partial, because see, this is basically the story of the partial pressure. Hemoglobin can bind with oxygen. It can also bind with CO2. It can also bind with CO. It depends on the partial pressure and its affinity. Now, here for the oxygenation to occur, right? For oxygenation, what do we need? What do we need for oxygenation? There should be high partial pressure of oxygen, and it is common sense. If partial pressure of oxygen is high, then obviously partial pressure of CO2 should be low. This is the first thing. Then, if there is high partial pressure, then students, it means that H positive concentration is low. H positive is low, right? Low H positive means high pH, because we know that high pH mane basic condition, low pH mane acidic condition, and during low pH, H positive is more. Now, other than that, even 2,3 bisphosphoglycerate, even it should be less, okay, and the temperature here is also low. So basically, what is high? Only one thing is high, and that is your, right, that is your partial pressure of, yes please, partial pressure of oxygen is more, CO2 is less, H positive is less, even the temperature here is low, and even your 2,3 bisphosphoglycerate is also low. So these are the conditions that favors oxygenation. These are the conditions that favors oxygenation. These are the conditions that are going to favor the, right, that are going to favor the oxyhemoglobin formation. Okay, right? So that's something you have to remember. That's something you have to remember. Are you getting my point? Now, how do we understand it? We understand it in this way, right? We will understand it by this oxygen hemoglobin dissociation curve. It will help us to understand this part. Are you getting my point? This oxygen hemoglobin dissociation curve will help us to understand this part. So this oxygen hemoglobin dissociation curve, just look at it. Look at the x-axis, it will tell you about the partial pressure of oxygen. Look at the y-axis, it is telling you about the percentage saturation of hemoglobin and oxygen. So the transport of oxygen, how can we understand it in a better way? We can understand it in a better way by studying the oxygen hemoglobin dissociation curve. Is that clear? Is that clear? Are you sure? So before going forward, I'll tell you some other facts also. You know, adult, in the case of adults, first of all, these factors are very important. You all need to, right? You all need to mark it. So the very first thing is that when you talk about the adult person, that adult person is having, so that adult person, a normal adult person, have 15 gram of hemoglobin per 100 ml of blood. Very important information, right? Per 100 ml of blood. I'm simplifying all the things from the NCERT, guys. Adult person, adult healthy person will have 15 gram of hemoglobin. You know that it varies in male and female, right? Male meat is 15 gram, but per 100 ml of blood. So basically, when we go for our hemoglobin test, so they do not check the hemoglobin in they basically check the amount of hemoglobin in 100 ml of blood. So adult person have 15 gram of hemoglobin per 100 ml of blood. So 1 gram of hemoglobin carries 1.34 ml of oxygen, right? 1 gram of hemoglobin. And so 15 gram of hemoglobin, which is in 100 ml of blood, and overall we have 4 to 5 or 5 to 6 ml of blood. So 15 into 1.34, so it is somewhere around 19.4 ml. So I can say that it is around 20 ml of oxygen. So per 100 ml of blood will carry near about 20 ml of oxygen or 19.4 ml of oxygen. So it is when we are not exercising, right? It is happening when we are not exercising. So normal physiological conditions may, right? Normal in during, so this is important, okay? So normal, under normal physiological conditions, under normal physiological conditions, what do you see under normal physiological conditions? You people will see that 100 ml of blood carries, I would say oxygenated blood carries 19.4 ml of oxygen, important information, and 100 ml of deoxygenated blood carries 14.4 ml of oxygen. So this is what we measure under normal physiological condition. 19.4 ml. Now you know how it is, because 1 gram of hemoglobin carries 1.34 ml of oxygen. So 100 ml of blood is having, if it is having 15 gram. So obviously I can change this data. I can ask the same question in the MCQ format. I can just simply tell you that if a person is having 12 grams of hemoglobin in its 100 ml of blood, then what will be the amount of oxygen that will be carried by 100 ml of blood or 1 liter of blood? That is how also I can ask the question. So you should be prepared accordingly. So 100 ml of deoxygenated blood carries 14.4 ml of oxygen. So what can we see here? So basically transport of oxygen by 100 ml of blood is, you see this, the difference is of what? 5 ml of oxygen. So this is what the 100 ml of blood is going to transport. Understood? 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 Right? So when you do strenuous exercise, like excessively you are exercising, when body is demanding more oxygen, so in that case what will happen? In that case 100 ml of blood can even transport 15 ml of oxygen, right? So it depends on the demand of our body. Okay? It depends on the demand of our body. So I believe this part is clear. Let's have a quick revision. We are talking about the transport of oxygen. I told you when oxygen and hemoglobin, right? Oxygen binds with hemoglobin. It is called oxygenation, not oxidation. So hemoglobin is the respiratory pigment. It's a quaternary protein having 4 polypeptide chains, 2 alpha, 2 beta chains. Globin is the protein part. Heme is the pigment here. So we have basically 4 heme and 1 hemoglobin, and it is having Fe2 plus, that is ferrous, right? So 1 Fe2 plus can join with 1 oxygen. So we have 4 heme and 1 hemoglobin. So 1 hemoglobin can join with 4 oxygen molecule. That is known as the formation of oxyhemoglobin. Now, for oxygenation, the partial pressure of oxygen is high. If partial pressure of oxygen is high, the partial pressure of CO2 will be low. H positive is also low, then pH will be high, 2,3-bis phosphoglycerate is also low, and the temperature is also low in that case, right? And then if I talk about a healthy adult, then adult male especially, that will have 15 gram of hemoglobin per 100 ml of blood, and 1 gram of hemoglobin can carry 1.34 ml of oxygen. So 15 gram of hemoglobin can carry 19.4 ml of oxygen. That's what you have to keep in your mind. So under normal physiological conditions, we will see 100 ml of oxygenated blood will carry this much of oxygen. 100 ml of deoxygenated blood will carry 14.4 ml of oxygen. So transport of oxygen by 100 ml of blood is what? It is 5 ml of oxygen. Understood? Understood? So this can change when body is demanding too much of oxygen when we are doing some exercise or not. Exercise. Now we will talk about the oxygen hemoglobin dissociation curve. That's what we have to discuss. A very important topic, and if there is any doubt, you know that you can freely ask me, right? So what's that? It is the oxyhemoglobin, oxygen hemoglobin dissociation curve, right? Oxyhemoglobin, oxygen hemoglobin dissociation curve. So that is what we have to study. Okay? So is it visible? Even if it is not, don't worry, we will share I'll share the notes in our official Telegram group. Is that clear? So here x-axis is telling us about what? So beta, it is a sigmoid shaped curve, right? It is a sigmoid shaped curve, right? It is a sigmoid shaped curve. So here this x-axis is telling us about the partial pressure of oxygen, that too in mmHg, and this side is telling me about the percentage of percentage hemoglobin saturation. Hemoglobin saturation means how much hemoglobin is attached to the oxygen, right? So percentage saturation of hemoglobin. Y-axis will tell me that, right? Saturation of hemoglobin. So this oxyhemoglobin या oxygen hemoglobin dissociation curve, what exactly it is, let's understand that first. Okay, let's understand that first, that what exactly it is. Okay, okay, so it's a graph which is plotted in between partial pressure of oxygen and percentage saturation of hemoglobin. And what exactly it is sigmoid curve, right? It shows us two shifts, left and right. It is sigmoid curve, or you can say that it is a S-shaped curve. Understood? Sigmoid curve, or it is the S-shaped curve, right? It is the sigmoid curve, and what else it is? It is the S-shaped curve. So before going forward, see this. Hemoglobin structure, alpha chain 1, chain 2, beta chain 1, beta chain 2. You have the iron here. This is your heme group here, and this is how the iron is. So Fe, it is Fe2 plus, that is your ferrous. This is the structure of the hemoglobin, number 1. Now, this is the curve that I'm talking about. Okay? So as I said, it is basically S-shaped. It is the sigmoid growth, a sigmoid curve, right? S-shaped it is. So very first thing, yes, I have to remember that what is there on the x-axis, and I have to remember what is there on the y-axis. Got that? Got that? Now, it will tell me how much oxygen and hemoglobin is saturated. I'll ask you one thing. Dekho, one hemoglobin can bind with 4 oxygen molecules. And I told you when the first oxygen molecule will bind, it will increase the affinity for the second and so on. So when one hemoglobin is attached to all the 4, so you will say it is fully saturated. It is fully saturated, means it is as per its efficiency. It has it is bind to all the oxygen molecules. Just let's suppose if your capability to study is for 2 hours, right? Then obviously in 2 hours you will be fully saturated, है ना? जैसे let's suppose if I will keep teaching for 12 hours or 13 hours, doesn't matter how good I'm teaching, there comes a time when you will start feeling saturated. You will be like, no, I cannot continue it further, right? I'm not able to do it. Yes or no? That I'm not able to do it. I cannot continue it further because I'm not able to understand anything. It happens. It happens when you attend classes continuously. You feel saturated. You are not able to manage. You are not able to understand anything later because you are saturated. You are full there, right? Just let's suppose even if the food is very good, but if your stomach is full, after a time, if you will force yourself to eat something, then obviously you know that you can even vomit. So it is like that. So what is the saturation? When one hemoglobin molecule is attached to the 4O2 molecule, it is fully saturated. Are you getting my point? What is it? It is fully saturated. It is fully saturated. So now here, this axis is telling me about the partial pressure of oxygen. This side is telling me about the percentage saturation of hemoglobin. Just say I'll give you the example. Let's suppose, just giving you a random example, and this is a fact also, that when the partial pressure of oxygen is 25 mm of Hg, 50% of the hemoglobin, right? 50% of the hemoglobin is saturated. Are you getting my point? Are you getting my point? Yes or no? Yes or no? Yes or no? Right? And do you know one hemoglobin carries near about 270 million, one RBC carries near about 270 million. Array, this is not work hard. Hemoglobin molecules. Do you know that? This is just for your knowledge, right? So one hemoglobin will carry, one RBC will carry that much of hemoglobins. Is that clear? Is that clear? Yes. Yes. So now imagine one can bind One can bind with what? One can. So this is in million. This is in million, not in billion. So one can bind with 4. So imagine in each RBC, if you have 270 million. So 4 into this 4 into 270 million. So it will be in billions, isn't it? It will be in billions, approximately 1.08 billion, right? Oxygen molecules will be attached here if all the hemoglobin in the RBC, they are saturated. Is that clear? Is that clear? So it is going to be approximately 1.08 billion, oxygen molecules per RBC. Just imagine, guys. So now, just say I have given you this example that at 25 mmHg, 50% of the hemoglobin is saturated. Now I'll give you a random example. Just say, let's suppose in one RBC, which is not true because you know the true data. Let's suppose 100 hemoglobins are there. है ना? Let's imagine that in one RBC, 100 hemoglobin molecules are there. So I am saying that when partial pressure is, let's suppose 5 mmHg, right? 5 mmHg. So 50 hemoglobin molecules are saturated. This is what I'm trying to tell you. So means 50% of the hemoglobin are saturated here when this is the partial pressure. So in general, this is what we study, that when the partial pressure of oxygen is around 25 mmHg, 50% of the hemoglobin got saturated. So we also call it as P50. We also call it as P50. So this curve is what? This is sigmoid. Now, here you have to understand one trick. You have to understand one trick here. What's the trick that you have to understand? You have to understand one trick here. The trick here is LARD. What is the trick here? The trick here is LARD. So what does it mean? Left means A for association. If you understand this, everything will be easy. Right means D for dissociation. That's what you have to keep in your mind. That's what you have to literally, that is what you have to keep in your mind. Left side, association. Right side, dissociation. That's what you have to keep in your mind. So ma'am, what's that? It's simple. Left side, association. Who's going to associate here? You're talking about oxygen, hemoglobin, na? So obviously oxygen plus hemoglobin. That is oxyhemoglobin. So you are, when the curve is moving to the left side, it is the oxyhemoglobin formation that is taking place. Their association is taking place. That is what we have to understand from this part. So this LA is telling me that left side is showing association, means when the curve moves to the left side, then oxyhemoglobin forms. Are you getting my point? Oxyhemoglobin forms when the curve moves to left side. When the curve moves to left side. Now understand this part, everybody. Those who think it's difficult, it is not at all difficult. You try to understand. Now you know what is on x-axis. You know what is on y-axis. Now what is happening next? When the curve moves to the left side, oxyhemoglobin form. And we know that what are the conditions required for it? Partial pressure of oxygen should be high, partial pressure of CO2 should be low, H positive should be low, it means high pH should be there, and obviously the temperature has to be low, and even your 2,3-bisphosphoglycerate that we also study during the glycolysis. So 2,3-bisphosphoglycerate, it should also be low. That is what we know. These are the conditions for the oxygenation. So if my curve is moving to the left side, so then obviously I have to be very clear that this is happening. This is happening. So this is actually the Haldane effect. Haldane's effect. Here you will see that saturation is maximum. Where is it taking place? Where the partial pressure of oxygen is high, that is your lungs. Are you getting my point? That is your lungs. Is that clear? Is that clear? So left shift, what this left shift indicates? So this left shift means what? Oxyhemoglobin formation. So it simply means more affinity for hemoglobin, less oxygen released. More affinity, less O2 released. Simple it is. That's the meaning of this curve. Are you getting my point? Are you getting my point? Done? Done? Any doubt here? Done? So when you talk about this curve, so this curve, right, this curve is having its, right, this curve, which curve? Oxygen, hemoglobin, dissociation curve. So its plateau region is in lungs, because in lungs you know that, right? That's why I'm writing it here. So its plateau region is in lungs. Why? Because here we know that there is a high partial pressure of oxygen. So even if there is even a slight change in the partial pressure, still the saturation is going to be maximum here. So in lungs, this is what you see. This is also we call it as the Haldane's effect. Is that clear? Is that clear? So here in the lungs, right, because the plateau region of this curve, I'll write it here. So here approximately 97 to 98% of hemoglobin is saturated. So in lungs you will see this shift. Are you getting my point? In the lungs, this is the shift that you all are going to see. Understood? In the lungs, this is what you are going to see. Any doubts? Any doubts? So 97 to 98% of the hemoglobin is saturated, right? 97 to 98% is saturation. So even if a partial pressure falls slightly, saturation, even if partial pressure of oxygen falls slightly, saturation remains higher. Are you getting my point? It will be higher. Okay, it will be what? It will be higher. Understood? Understood? Sure? So this is going to ensure maximum loading, of oxygen in lungs. So in lungs, we will see the curve is towards the left side, and we'll also remember it. It's the Haldane effect. We'll see it when I'll teach you the CO2 release. Right? We'll teach you the CO2 release. Is that clear? Sure? Sure? Okay, so the steep region of this curve, where will you see the steep region of this curve? You will see it in the tissues, where the partial pressure of oxygen is also low, 25, 30, like this. And even if PO2 falls slightly, a large amount of oxygen release occur. So this condition here, right, the steep curve here in tissue, here you'll see that. So here it ensure maximum unloading. जैसे here loading, ensure maximum unloading of oxygen in tissue. These are the conditions that are going to do that. Right? These are the conditions which are going to do this here. Is that clear? So here partial pressure of oxygen is quite low, 20 to 30. So small fall, if there is small fall in PO2, large release of oxygen will occur. Right? So this is about the curve, right? That where do you see this curve is, in plateau region and steep region. So obviously here also, look at this part. Look at this large part here, right? Look at this large part here. So I told you this thing, that L means A, means left side means association. So left side mein if association is occurring, if oxyhemoglobin is forming, these are the conditions which are required. So right side is dissociation. So opposite to that, partial pressure of oxygen should be low, partial pressure of CO2 should be high. So H positive concentration here, it should be high. So it means acidic pH is there, low pH is there. Moreover, temperature here is going to be high because high metabolism is there. And here 2,3 bis phosphoglycerate is also high. So you just have to right? The opposite of that here. So these conditions will result in dissociation. So here oxygen dissociate from hemoglobin. Here oxygen dissociate from hemoglobin and is transported to cells, and which is important. So what is this? This right shift is basically what? It is the Bohr's effect. B for breakup. That's how you can remember. Right? That's how you can remember. So what is the meaning of B here? B for breakup. Whose breakup we are talking here? We are talking about the breakup of oxygen and hemoglobin. So how the breakup of oxygen and hemoglobin will be there? So hemoglobin is shrewd here. Though it is not shrewd its job, but okay, to understand this, hemoglobin is shrewd here. Hemoglobin is like, oh, now the CO2 partial pressure is high, I should go for CO2. Why to stay with the O2? So hemoglobin is mean here. Hemoglobin is like, if partial pressure of oxygen is more, I'll go to oxygen. If partial pressure of CO2 is more, I'll go to the CO2. So obviously, if hemoglobin will go to the CO2, there will be the breakup of oxygen and hemoglobin. How will it occur? We will discuss it further. But this right shift is due to the Bohr's effect. So B for Bohr's, B for breakup. Whose breakup are we talking here? We are talking about the breakup of oxygen and hemoglobin here. So this is dissociation. So this is the Bohr's effect, right? And this is very important. So this Bohr's effect is responsible for the transport of oxygen. So you see this. I told you this oxygenation is temporary. It is reversible. You imagine if hemoglobin, now I am just showing you the bad side of the hemoglobin, that what hemoglobin is doing. Hemoglobin is just attached to the, what do we say? It is attached to the, oxygen is attached to the hemoglobin. If they will stay together, imagine. If hemoglobin, if it will not release oxygen, if it will stay with oxygen always, then how our cells are going to use that oxygen? How our cells are going to work? Our cells, they need oxygen for the oxidation of the glucose. They need it for the respiration, for the cellular respiration. But if hemoglobin will not leave it, then how will you get, how the cells will get it? Same, just let's suppose if somebody is having your mobile phone for the delivery, but if that person will not give it that to you, then what will you do? What will you do? That thing belongs to that person temporarily. It belongs to you ultimately. So hemoglobin and oxygen, this association is important, because hemoglobin, it needs to carry the oxygen to the tissues, to the cell, right? Where the metabolism is high, where the CO2 is high, where the demand of oxygen is there. So in that tissue, due to the high partial pressure of CO2, even we know that metabolism is high, that's why partial pressure of CO2 is high. So obviously that will be just like a breakup for the oxygen and hemoglobin, this breakup is much needed. Then only oxygen will leave hemoglobin, then only oxygen will come to the cell, and cells will be able to do the respiration. So this is about the shifts, this is about the curves. I hope you are able to understand it. You can see it, I have added it here. I have mentioned everything here, guys, and this is what is given here. So here in the NCERT, I have simplified your NCERT, I have clarified your NCERT. Look at the NCERT. They have simply mentioned that this curve, right, these are the factors like partial pressure of CO2, high constant, H positive concentration. They will tell us that how, all that things are affecting the binding of oxygen and hemoglobin. In the alveoli, where there is high partial pressure of oxygen, low PO2, less H positive, low temperature, right, the formation of oxyhemoglobin occurs. So I told you this is the plateau region of this curve, oxyhemoglobin forms. This is the Haldane's effect as well. So low PO2, high partial pressure of CO2, high CO2, H positive concentration, they are for the dissociation. So oxygen gets bound to hemoglobin in the lung surface and gets dissociated at the tissues. We know it very well. So every 100 ml of oxygenated blood deliver 5 ml of oxygen. So in NCERT, they have just mentioned this, and I have explained you how. So this is the curve. This is the oxyhemoglobin dissociation curve here. So this is all about this. Now the next topic is the transport of CO2, which is again a very, very, very interesting one. Okay. This is what we have to discuss. Ready? Ready? Beta, I don't know about the duration. We will finish it and then we will say bye-bye to each other. Ready? So in the tissue, the curve will shift to the right side. In the lungs, the curve is towards the left side. Now let's talk about the transport of CO2. So if there is any doubt, then please tell me. Done, students? Okay, so one more thing, your oxygen. Oxygen doesn't oxidize hemoglobin. Do you know that? But ozone can oxidize hemoglobin. That's why these gases are very dangerous. So oxidized hemoglobin is called methemoglobin. Understood? That is methemoglobin. Chaliye, next is the transport of CO2. This is again very interesting. There are three ways by which CO2 can be transported, right? So CO2 ka transport is in the form of, so in dissolved state. And we know that its solubility is more. So 5 to 7% of CO2 is transported in the dissolved state. In the dissolved state. Am I clear? In the dissolved state. Number 2, in the form of carbaminohemoglobin. In the form of carbaminohemoglobin. So near about, see, carbaminohemoglobin is the word here, right? Carbaminohemoglobin is the word here. So this is something very important, right? This is something very important. See, in the dissolved state as in plasma, okay, that is easy. 20 to 25% of CO2 is transported in the form of carbaminohemoglobin. In the form of carbaminohemoglobin. Are you getting my point? What is this? Dekho, your CO2. When your CO2 binds with, this is important, please pay attention. When it binds with hemoglobin, guys, when your CO2 binds with hemoglobin, then it is forming what? Carbaminohemoglobin. Why am I emphasizing it? I'll tell you. I have seen students making this mistake very commonly. But when CO, that is carbon monoxide, binds to hemoglobin, then it is carboxyhemoglobin. I have seen students making this mistake so many times. So many times I have seen people making this mistake. You please avoid it. So when CO binds to the hemoglobin, देखो, when CO2 binds with the hemoglobin, that is carbaminohemoglobin. But when your carbon monoxide, we know that carbon monoxide, even those who smoke, that smoke is after cigarette smoking also, that smoke contains the carbon, this monoxide, right? And we should not, so this is binds to. So when this binds to the hemoglobin, it is carboxyhemoglobin. And do you know CO, carbon monoxide affinity is too high for the hemoglobin than the oxygen or even than the CO2. Matlab carbon monoxide is, its affinity for the hemoglobin is very, very, very high. If there is CO in your blood, right? Your hemoglobin will prefer to bind with the CO. So your body cells, tissues, they will be deprived of oxygen, and you know that then what can happen. So it is very dangerous. That's why it's not good to smoke. Smoking is really very bad. Okay, right? So do remember it. So carbaminohemoglobin will form. Carbaminohemoglobin will form when CO2 plus hemoglobin is attached, right? And carbon monoxide will bind to the hemoglobin, then it is the carboxyhemoglobin. So do keep it in your mind. And the third is in the form of bicarbonates. Are you getting my point? In the form of bicarbonates. So maximum, right? 70% or above, 20, 25, 70, around 70% I would say or above is transported in the form of bicarbonates. In the form of what? In the form of bicarbonate. So this is something which is going to be very, very interesting now. And for this interesting part, I have a diagram, right? This is what you have to study. Trust me, it is going to simplify it. Dekho, this is your RBC. Firstly, I'll explain it with the help of this diagram. Then, beta, I will write everything. This is RBC. You know one thing. When we talk about blood, we know that blood is having two components. Blood is having plasma and blood is also having formed elements. Plasma is more, formed elements are less, around 55%, 45%. Formed elements means you are talking about the cell. We are talking about the RBCs here. And we know about the plasma, that pale yellow color liquid in which these formed elements are present. Now, imagine it in this way. This is your plasma. Here you have RBCs. This is your plasma and here you have RBC. Now we have to talk about an enzyme which is known as carbonic anhydrase. What's the name of enzyme? This is carbonic anhydrase. Carbonic anhydrase. And let me tell you, it is the fastest enzyme. Let me tell you, it is the fastest enzyme that we know. Slowest is ATPase, fastest is this one. Fastest is this one, right, beta? Beta Sandra, you please spam more. You please spam more, right? Keep spamming, don't worry. Now, so definitely I will not upload the remaining portion of human health and disease. You keep spamming. I will definitely not upload it. Done. So this is the fastest enzyme. Now, plasma contains this carbonic anhydrase, but its amount is negligible. But its amount is what? It is negligible. Very, very less. Are you getting my point? In RBC, you will see that carbonic anhydrase is present. Right? Here it is in excess in comparison to plasma. Here it is in excess in comparison to plasma. So what is the function of this particular enzyme? This enzyme is having a very simple function. See, this is our RBC. In the surrounding, we have the plasma. So when there is this carbon dioxide is present. That's what we have. Now I'll tell you whatever conditions we have discussed, we will also talk about that. Dekho, carbon dioxide and water in the presence of this carbonic anhydrase, they will join and it will form the carbonic acid. What will it form, beta? It will form the carbonic acid, that is H2CO3. Can you see that? H2CO3. Is that clear? Is that clear? Is that clear? This carbonic acid, though it's a weak acid, again, the same enzyme, the fastest enzyme, it is dissociating it into bicarbonate. This is the bicarbonate and this is, you can see, H positive. Now you'll understand when we talk about the transport of CO2, we say na high partial pressure of CO2 will result in high H positive concentration. Now you guys can see the reason here that why during CO2 high partial pressure of CO2 why the H positive is more. Now what this H positive will do? What is the fate of this? I told you I'll write it later, but I'm writing here only. So what will this H positive do? It will combine with hemoglobin to form hemoglobinic acid, right? Right? Yes or no? Right? This H positive, it will combine with hemoglobin and form hemoglobinic acid. This is what they are going to form. Imagine. And do you know hemoglobin act as a buffer? You know, na buffer, it will maintain the pH. If it is becoming too acidic, then it will make sure that it should reduce, right? Even if it is becoming more basic, so it maintains it, right? It maintains it. So this H positive here, it will combine with the hemoglobin and form the hemoglobinic acid. And now obviously it is going to dissociate the oxyhemoglobin as well, right? Will reach there one by one. So this is the fate of H positive, that this H positive will do what? It will combine with the hemoglobin, right? It will combine with the hemoglobin and it is going to form hemoglobinic acid. And hemoglobin, I told you already, it acts like what? It acts like buffer. Now what's the fate of this bicarbonate? We know that in the plasma, very minute amount of CaH present. So in the plasma also, beta, we have bicarbonate, but it is in very less amount. Bicarbonate is still there, but it is in very less amount, right? Right? Now what will happen? It's very simple. This bicarbonate will be like, I want to go out. Throw me out. I want to go out. Bicarbonate is like, I am too much in RBC. I want to go out in the plasma, right? I just want to move out in the plasma. But if that thing will occur, you know, it will disturb that balance. Then what will happen? Bicarbonate from RBC, it will move to the plasma just to balance that thing, just to maintain that electrical neutrality. Chloride from the plasma will enter here within the RBC. So this is basically chloride shift, not given in NCERT, but a very, very, very important topic. So this is chloride shift, also known as Hamburger phenomena. Also known as Hamburger phenomena. So what is happening here? Bicarbonate from RBC moves to plasma and chloride from plasma enter RBC. Chloride from plasma. So bicarbonate from RBC moves to plasma and chloride from plasma enter RBC. So this is your chloride shift. This is chloride shift. Are you getting my point? This is what chloride shift is. This is chloride shift, also known as Hamburger phenomena. Firstly, tell me this part clear, then I'll tell you the next portion. Any doubts here? Any doubts there? Done, beta? Sure? Done, beta? So next is done. So now this bicarbonate combined with sodium in plasma and form NaHCO3 means sodium bicarbonate, right? So this is going to form sodium bicarbonate. That will happen in plasma, right? So now this HCO3 negative combine with Na positive in plasma and form sodium bicarbonate. Now this blood is deoxygenated, of course. It will be carried to what? Obviously to the lungs. And moreover, at high partial pressure of carbon dioxide, right students? So moreover, at high partial pressure of carbon dioxide, we know that CO2 reacts with hemoglobin, isn't it? Yes or no? Yes or no? It will form carbamino hemoglobin, and we know that it is something which is unstable, so we know it very well. Now what will happen? This deoxygenated blood, it will pass to the lungs. Yes or no? Yes or no? This will pass to what? This will pass to the lungs. Why? Because now obviously CO2, it needs to release as well. So now I'll tell you, there is something called Haldane's effect. I already told you what are the conditions for that, but this Haldane's effect is very important. Why it is very important? Because this Haldane's effect, it will explains the transport, or I would say release of CO2 from the body, right? So this is what we have to discuss now. So up to this part all clear? Up to this part all clear? Look at the diagram here. Then they go CO2 plus H2O, carbonic anhydrase will form carbonic acid. Then it will dissociate. H positive will combine with hemoglobin, form hemoglobinic acid. You know that hemoglobin acts like buffer. So this hemoglobin, so obviously, right, when H positive is increasing, you know that it is also a favor that dissociate oxyhemoglobin. Partial pressure of CO2 is high here, that also dissociate oxyhemoglobin. So you can see oxygen is getting released. Hemoglobin, this hemoglobin is now joining with the H positive. Hemoglobinic acid is forming, and this oxygen here is going to the tissue. Bohr's effect, right? Now this bicarbonate is moving to the plasma. Just to maintain the plasma neutrality, chloride is moving here. This is the chloride shift hamburger phenomena. Now when we move to this part, so this is happening in the tissue. Now come back to the lungs, where Haldane's effect will be applicable. So in the lungs, you people know, partial pressure of oxygen is high, partial pressure of CO2 is less, H positive is less, temperature is also less, even 2,3 bisphosphoglycerate is less. Now in the here, what's going to happen? Due to these conditions, a reversal will occur. Here you will see in the lungs, there is the reverse chloride shift. Do you know that? Reverse chloride shift occur, like bicarbonate from RBC. Jaise initially it was moving from RBC to plasma. Now bicarbonate from plasma is moving into the RBC, chloride from RBC moving out, right, into the plasma. And because partial pressure of oxygen is high here, so oxyhemoglobin will form, which is a strong acid than the hemoglobin. And obviously due to that, HHB means hemoglobinic acid will dissociate. See, H positive is free. This will join with this bicarbonate again. This will form carbonic acid, and then again CO2 plus water, and this CO2 will be released out. It will be exhaled out. So in the lungs, reversal of what we studied will be taking place. So here again you can see this. So in the lungs, reverse chloride shift is there. In the lungs, oxyhemoglobin form, that is stronger acid than the hemoglobinic acid. So hemoglobin will release H positive. That H positive will join again here with the bicarbonate. Carbonic acid will be formed. That carbonic acid will again dissociate to CO2 and water, and the CO2 is released. So this is the Haldane effect that is helping, right, for the release of the CO2, and Bohr's effect for the release or the transport of the oxygen. Is that clear? Is that clear? Sure? Done, beta? Sure. So release of CO2 in lungs. So a series of reverse reactions occur in lungs. Done, beta. A series of reverse reaction will occur in lungs. I have already explained you this. Okay, beta. So just write down. So oxygenation of blood, right? So you can write it. Oxygenation of blood, of hemoglobin, cause, even blood also you can write, release of H positive from hemoglobinic acid, True that? Right? Right? Now, this H positive combine with bicarbonate formed due to the breakdown of sodium bicarbonate. And why is it so? Why is it so? Because due to high partial pressure of oxygen, low partial pressure of CO2, right? Low H positive, right? Low temperature. We know that all that things are favoring these reversal reactions. So H positive will combine with bicarbonate. So from where we got this bicarbonate? Because we know that, right? Because we know that there is the breakdown of this. Is that clear? Is that clear? Done? Done? Yes, please. So now next we know that carbonic anhydrase combine this H positive and HCO3 negative, form bicarbonate, then forms CO2 and water. Moreover, due to high partial pressure of oxygen, we know that carbamino hemoglobin dissociate and again dissociate. So that's how these conditions causes release of CO2 and then its exhalation. Understood? Then its exhalation. So that is taking place. So already explained you this and now I am mentioning it in the form of points here. Got that? Done? Is that clear? Sure? Okay? Done? So binding of, so this is what your Haldane effect is, already explained. So writing this again so that you can remember this. So binding of oxygen with hemoglobin displace CO2. Understood? It displaces CO2 from blood, right? It displaces CO2 from blood. This is the Haldane effect. This is the Haldane effect. Am I clear? So guys, this is the Haldane effect and we know that oxyhemoglobin is stronger acid than hemoglobin, right? Yes or no? It is a stronger acid than hemoglobin and thus displays CO2 easily. Okay? Done? So that's a PYQ, just keep it in your mind. So Bohr's effect is important to displace oxygen from hemoglobin. Your Haldane's effect, which is taking place in the lungs, displace CO2. And then obviously, overall you can say that this Bohr's effect is important for the transport of oxygen to the cell. Haldane's effect is important for the transport of CO2 from the lungs to the outside. So that is what you have to remember. So this is what you need to know about this topic. So I have explained this topic in depth, have added these diagrams for your better understanding. If you still struggle here, you can just read about it, and then later you can just tell me in the comment section, guys. I will definitely make a short video on that too. So remember, under normal conditions, guys, under normal condition, जैसे 100 ml of the blood carries transport 5 ml of the oxygen. So 100 ml of the blood transports 4 ml of the CO2 to the alveoli. So this is again important information. Understood? This is again important information. So this is all about this. So last, or I would say the second last topic is the regulation of the respiration. So here I'll move very quickly here, right? So yes, please. So here you guys can see human beings, they can moderate the respiratory rhythm to suit the demands of the body tissue. When we do the exercise, we need more oxygen, right? So obviously body adjusts itself according to that. So a specialized center is in the medulla region. So where is the medulla? It is in the hindbrain, right? So it is the respiratory rhythm center which is there in the medulla oblongata. It is primarily responsible for this regulation. It's a repeated previous year question, guys. Got that? There is one more center. It is in the pons varolii region of the brain. It is pneumotaxic center. Right, so respiratory rhythm center in medulla, this pneumotaxic center in the pons, it moderate the function of respiratory rhythm center. So the neural signal from these centers, they can reduce the duration of inspiration and alter the respiratory rate. So what you have to understand, this is something you have to keep in your mind. If in the arterial blood, it is not the oxygen that regulates the respiration, it is the CO2. If I'm writing this line here, so do you never forget, if CO2 increases in the arterial blood which is supposed to carry what? Oxygenated blood. If CO2 partial pressure increases there, then it sends a signal for inspiration. This is actually a signal for inspiration. It is not the oxygen. It is the CO2 that is going to decide that. It is the CO2 that is going to help there. Is that clear? So a chemosensitive, which is sensitive to the chemical, it is situated near to this rhythm center. This is highly sensitive to the CO2 and hydrogen ion. So if there is any increase in this, the center will get activated and the adjustments will be done, students, right? Right? So increase in these substances can activate this region. Okay, beta. So receptors associated with aortic arch and even with the carotid artery, which will give the oxygenated blood to brain, they also recognize these changes in these two, right, and send the necessary signal. So I told you it is the role of oxygen in the respiratory rhythms is quite insignificant. So where we have to see the CO2, be clear, CO2, if CO2 and H positive increase, where? In the arterial blood, right, in the arteries, in the oxygenated blood, right, not in the venous blood, arterial blood, then it will send a signal. Got that? So little more of it. I've added a flow chart. चलो, we'll share it later. हाँ, नहीं है, ये देखो. So here you guys can see. देखो, in the medulla region, dorsally and ventrolaterally, what do you see? Dorsally in medulla, you have dorsal respiratory group, short form DRG. This mainly causes, जैसे medulla is having respiratory rhythm center, so dorsal side is having dorsal respiratory group, right? Then mainly causes the inspiration. It will send the signal to inspiratory muscle for about 2 seconds, which will, inspiratory muscles will be contracting for 2 seconds, silent for 3 seconds, which contract causing inspiration. Then ventral respiratory group, beta, ventrolaterally in medulla. It sends signal for both inspiration as well as expiration. So it is active when respiratory activity increases, and this is the one, this ventral respiratory group is the one that cause forceful inspiration and expiration. Now in Pons Veroli in the NCERT they have mentioned this pneumotoxic center. So this is the one that signals to the dorsal respiratory group. Dorsal respiratory group, it will send the signal, and inspiratory neuron of the ventral respiratory group, that now, right, that will limit the respiration. It keeps a check on it. Just mummies, M for medulla, M for mummy. Mummy is soft. Mummy is like, "Arey, my baby needs 200, 500. Chalo, I'll give you." But Papa will do. Why? Why are you giving the extra money? Why are you giving the money without requirement? Right? They are keeping the check. Right? Are you getting my point? So that's how it works. So dekho, so send signal to dorsal respiratory group and the inspiratory neurons of ventral respiratory group, that you should check, you should limit the inspiration by controlling the switch of point of inspiratory signal. So this pneumotoxic center will act like, it will act like a check on this respiratory rhythm center for the inspiration. Then apneustic center is also there. It is in the lower part of the pons. It is also important. So in association with this pneumotoxic central, it controls the depth of inspiration. Depth of inspiration. Is that clear? Right? Is that clear? Done? Okay, so this is all about these centers: pneumotoxic, apneustic center, dorsal group neuron, ventral group neuron. So I hope this is clear to all of you. And when we talk about the disorders, so it is something that you guys can read, but still, types of hypoxia, hypomanyless, oxia, oxygen. So hypoxia, partial pressure of the arterial blood is low. Anemic hypoxia, where amount of hemoglobin is low, so carrying oxygen is low. Stagnant or ischemic hypoxia, blood flow to the tissue is low. Or histotoxic hypoxia, tissue cannot utilize the oxygen. So disorders overall are very important. I always take a short video on that. Then asthma. So asthma is difficulty in breathing, right, causing wheezing due to the inflammation of bronchi and bronchiole. So what sort of MCQ can come in asthma? Dash, inflammation of dash and dash occur, bronchi and bronchioles. Emphysema due to continuous cigarette smoking, and it can be treated by giving alpha-1 antitrypsin. In biotechnology, I have told you this. Alpha-1 antitrypsin, because it will stop the trypsin activity. So it's a chronic disorder. Alveolar walls are totally damaged here, right? Respiratory surface due to which it will decrease. So basically in this case, what will happen? The fibrous tissue will be more, its proliferation increase. The lungs, they will lose their elasticity. Are you getting my point? Lungs will lose their elasticity. So this is the major cigarette smoking is the major cause. So it is treated by giving alpha-1 antitrypsin. Understood? Baki occupational respiratory disorder, like some people work in that areas where too much pollutants are there, just asbestos, silica. So silicosis, asbestosis can occur. So in certain industries, especially those where the stone breaking or grinding is there, right? So dust will enter in the body will have the protective mechanism to cope up with that. Body will do what? Will proliferate. The proliferation of fibrous tissue again will occur here. Inflammation will also be there, right? And thus causing serious lung diseases. So this is it. So there are others also, but yeah, this is all about it. Baki, we know that mountain sickness or altitude sickness, when people from plain regions, they go to the higher altitudes, you know that they are not able to adjust immediately. They have to acclimatize their body. So to acclimatize, the body start making excess of RBCs, right? The curve, the affinity, what will happen? The curve shifts more towards the right side there, right? More RBCs are there. The affinity for the hemoglobin and oxygen is there. But yeah, basically what is there? Oxygen and hemoglobin, its dissociation occurs at a faster rate, so that all the body parts can get the oxygen. Is that clear? Is that clear? That? Okay. So this is all about it. And guys, it's not like that I'm going out of NCERT or in NCERT. Whatever I have explained to you, this is much required for your exam. This Haldane effect, this chloride shift, these terms are not used in your NCERT, but you check the last 10 years PYQs, you'll get the question from that part, right? So study properly, right? Study properly and do not miss the most important units. Study them sincerely, right? Today we solved some questions also. Rest you can get the practice sheets, right? Baki here you will see some extra questions are also there. So I'll upload the notes. Okay, I'll upload the notes, and in the description box of this particular video, right, in the description box of this particular video, you have the link for the practice sheets and for the notes as well that you guys can access. So keep studying, keep revising. Okay, breathing and exchange is done. So next chapter is going to be locomotion and movement. Done. So next chapter is going to be locomotion and movement. So that is it. Take care, study well. If you like the session, do let me know in the chat section. So guys, I also upload shorts where we discuss the tricks, the important questions. So do subscribe to our channel if you are new to our channel. So take care, bye bye.